We are unable to
national
see.
and
Excuse me, Dr. Aire, we are unable to see your
ah,
slides.
okay. Now his slides are visible.
Yeah, now it's
Yes,
visible.
sir. Yes, sir.
Okay. So total, I have published 55 national and international papers. Cumulative impact factor of these papers is 81, and average impact factor is 1.47. I have attended and completed 23 different workshops and training programs. Attended 55 national, international, and conferences and webinars. 77 abstracts have been published in different abstract books of conferences, seminars, works, and symposia. I've been worked as a resource person or delivered the lectures in 19 different events. I have participated in organization of workshops, seminars, and conferences. About 9 different events are being organized at Yeshwantrao Chavan Institute of Science, Satara. Currently, I am working as associate editor of Plant Molecular Biology Reporter Journal, which is published by Springer Nature, and served as reviewer for more than 100 research articles across more than 25 peer-reviewed journals, including Vegetos. I am a regular reviewer for the working as a regular reviewer for Vegetas also. So this is about what research work has been carried out. Now I am only highlighting some of the research projects which we have been completed or in progress. So my area of specialization is particularly plant tissue culture, stress physiology, and crop wild relatives, particularly phytochemical analysis of the crop plants or wild relative of crop plants, and some medicinal plants also. So we have working on finger millet stress tolerance and application of silicon for retaliation of salt and rot stress tolerance. So silicon has found to be a beneficial element on osmotic stress tolerance in many of the plants. So we worked initially we worked on the finger millet, and we have got very good results, and we have described for the first time about the silicon transporter genes in finger millet. And nowadays currently we are working on all 8 millets. So apart from the finger millet, we are also working on the foxtail millet and other small millets also, like kodo millet, proso millet, and all that work is currently in progress. So we have a very good research outcome of this project is having very good number of research papers in reputed journals also. Then in some medicinal plants, we have analyzed the phytochemical constituents, like in the berries of sandalwood, that Santalum album, and we have published that paper in Biocatalysis and Agricultural Biotechnology. Then we have working on the endemic plant Freria indica, which is endangered and endemic in the western Ghats of
Hello.
Maharashtra. Hello.
Now,
Hello.
Sir, you
So,
can continue, sir.
Okay. So that paper is also being published in Biocatalysis and Agricultural Biotechnology Journal. Then we have moved to the crop wild relatives, particularly in Vigna, in genus Vigna, section Aconitifoli. We have worked on all 4 wild relatives of Vigna Aconitifolia, that namely Vigna trilobata, Vigna indica, Vigna stipulacea, and Vigna khandalensis. So we have analyzed the phytochemical constituents from all the wild relatives we have collected from different localities. We have made the comparative analysis, And then after that, we have worked on the stress tolerance capacity of the crop oil relatives, and we have found that Vigna stipulacea is found to be a good plant, good member to for the crop improvement. And now nowadays we are continuing with the Vigna stipulacea, particularly in polyploidy induction, and then we are going to the stress tolerance studies in the polyploid plant. Earlier, I have worked on plant tissue culture in Bacopa monieri. So we have standardized the tissue culture protocol. Then we have worked on the in vitro stress tolerance using NaCl, KCl, and calcium chloride also. And we have transformed the VPPS gene in the Bacopa monieri to check the stress tolerance capacity. We have found very good results, and we are also promoting this particular plant as the model plant to study the transgenic studies, as it gives rise very good transformation efficiency. So we have worked on that also. Then we have worked on the capacity of the plant for textile dye degradation also, and we are getting very good results in that aspect also. So in the future plans, we are working on some image processing using AI and machine learning in botany, particularly in grapes, grape diseases. So biotic tolerance also. We are trying to have the field-based learning and project-based learning as executed by the National Education Policy 2020. So we are also working in that direction also. Recently, we have our first batch had completed their third year under the NEP 2020, and we have almost sent all our BSc 3 students and MSc students for their on-job training and field projects and community engagement program also. So we are working in that particular direction also. In terms of research, I'm continuing my research in stress physiology and crop resilience. So we are integrating the omics approach with the help of Department of Botany, Savitribai Phule, Pune University, Pune. Now I am started working on silicon nanoparticle and their interactions under stress tolerance. We are continuing the phytochemical analysis and secondary metabolite studies in crop oil relatives, as well as in the medicinal plants. And in plant tissue culture, we are working on the polyploidy induction, particularly in Vigna stipulacea. So finally, thank you all for patience hearing, and I'm again thankful to the authorities of Society for Plant Research for providing me this opportunity. So I'm completed with my presentation. If you have any questions, please you can have.
Thank you. Thank you, sir. Thank you so much for your insightful and valuable presentation. Now moving towards the second participant, may I now invite Dr. Saurabh Pandey, sir, from IGKV Raipur, to kindly deliver his lecture. Sir.
Thank you, ma'am.
I think Professor Nana has got some question. Yes, Professor Nana. Yeah, kindly unmute your mic.
Sir, your mic is muted, sir.
Professor Nana, it is still mute. Okay, it is. We can take up
Now
later
it is
on. Yeah, it is okay.
Yeah.
It is okay now. Please ask.
So then, where did you get these wild relatives of Vigna species?
Sir, we are having 25 accessions from NBPGR, New Delhi, and others 20 accessions of Trilobata indica and Stipulaceae and Khandalensis. We are collected from Tamil Nadu. We are collected it from the Maharashtra also. So from Madurai, we have the accessions of Vigna Trilobata, particularly Vigna Trilobata from Tamil Nadu district, Tamil Nadu state.
So regarding polyploid induction, what type of methods you are using?
Currently, we are
polyploid.
using colchicine treatments. We are inducing the polyploid, and now we are in the testing phase of that analytical confirmation of the polyploids.
Did you get any plant for polyploiding?
Right now, we have the plants, tissue culture plants, in vitro plantlets, and we have confirmed it with the flow cytometry. But still, we are want to confirm by conventional cytological techniques. So we are waiting for that also. For that.
Did you analyze any biochemical, physiological, and morphological?
Not still not. After confirmation, we are going with the stress tolerance experiments.
Okay, then nice. Okay, thank you.
Thank you, sir.
Okay, sir. Can we call upon now, invite Dr. Saurabh Pandey, sir?
Thank you, ma'am. Can I share the slides, ma'am?
Yes, sir. Yes, sir. You can share the slides.
Slides are visible, ma'am?
Yes, sir. Now your slides are visible.
Okay. Thank you very much. Good afternoon, everyone. First of all, I would like to thank Society for Plant Research, Professor S.K. Bhatnagar, sir, Vanna Bose, ma'am, and it is my honor to accept the SPR Fellow Award. And my lecture is today titled as Resilience at the Molecular Level, where I will discuss my journey, how I am integrating plant stress biology with genomics and molecular biology. So basically, I am integrating 4 different aspects of plant biology, where I am dealing with plant molecular biology, with the experiments of gene expression, signal transduction pathways of hormones and protein functions, along with stress biology, where I have worked upon heat stress, drought stress, salinity stress. Then the functional characterization of the genes responsible for giving the stress tolerance. It is characterized through different techniques like transcriptomics, genome sequencing. I am also working with the gene editing technologies, and we have worked with the virus-induced gene silencing that is also can be used for functional genomics studies. Then the 4th pillar of my study is the plant-virus interaction. This is the fundamental work that I have done during my PhD studies, where I have studied about tomato leaf curl New Delhi virus interactions, and I have characterized one RNA helicase for its viral pathogenesis and how it is giving the defense. So coming to my academic journey, till now I have around 54 research and review publications. I have also edited 7 books, written 14 book chapters, and as you know that I am from agriculture background, so the foremost work is popularize our technology to the farmers. So for that we have written the 15 popular articles. Apart from that, I have done my undergraduation from the Indira Gandhi Agriculture University itself. Then I moved to University of Agriculture Sciences, Bangalore, for my master's in plant biotechnology, where I have got the university gold medal. Then I have qualified the ICR exam for SRF, and CSIR JRF as well as DBT JRF, through which I have got admission into the National Institute of Plant Genome Research for my PhD studies. So after that, I have joined as assistant professor in Guru Nanak Dev University, Amritsar. Then I have moved to Indira Gandhi Agriculture University on October 2024. During my stay in Guru Nanak Dev University, I have got the SERB Startup Research Grant, which is now ANRF Prime Minister Early Career Research Grant. I have also awarded different Young Scientist Award from different societies. I am currently life member of Indian Virological Society, Indian Academy of Horticultural Science, Society for Plant Biochemistry and Biotechnology, and I recently joined as a life member for Society for Plant Research. So starting from my foundational work that I have done at NIPGR New Delhi, so which is conducted under the mentorship of Dr. Manoj Prasad sir. So here I have characterized one RNA helicase, that is dead box RNA helicase from tomato, SLD35, against the Gemini virus, tomato leaf curl New Delhi virus, which is a bipartite virus. Basically, if you know, virus has two different functions for hijacking any host plant. It has to carry out its replication, so it will hijack the host machinery for replication, and it has to protect its nucleic acid from the different host defense mechanisms. So what protein are the two most important proteins that we can target for giving the tolerance? So based upon this study, this is my PhD title, Deciphering the Role of RNA Helicase upon Tomato Leaf Curl New Delhi Virus Infection. So in this study, what I have got, the RNA helicase, which is subjected for characterization, I have characterized it through the virus-induced gene silencing as well as transgenic studies. So upon characterizing this gene, we have got that this gene is interacting with the coat protein of virus. Upon interaction with coat protein, the AC2 gene could not bind to the virus replication machinery. So in that way, the virus replication is also got inhibited, as well as its movement is also restricted. So in this way, tomato leaf curl New Delhi virus is restricted by the overexpression of RNA helicase, that is SLD35. So while characterizing, I have used the virus-induced gene silencing, where in preliminary studies we have got that by silencing this gene, the viral titer and expression of certain genes got overexpressed. So then I choose the characterize this gene, and by transgenic expression, I have got this model. So based upon this study, I have got a few papers in environmental and experimental botany, as well as in plant science. This work is very much covered with the media in the down to earth. I also got interviewed with the Rajya Sabha TV and Science Monitor for highlighting this work. The interesting thing was that while my main objective was that I have to characterize the gene against the virus, but during the study, the overexpressed plant also was tolerant against the heat stress. That's why this work was very much covered in different news articles. So apart from that, in my early career, I have also worked on the seed priming aspect of plant biology, where I have moved from genomics to the practical physiological applications. So these we have applied different salts and checked the biochemical responses, how they are giving the stress tolerance. So this work is also published in different good journal like Plant Stress for abiotic stress tolerance. So seed priming, by seed priming, we can induce the drought as well as salinity priming for different plants. So Recently, we are working on different phenotyping methods that we can standardize for different plant stress. I am also working on different hormonal interactions. So how this is, they are giving the different stress tolerance by spraying and everything. Apart from that, I am also started the genome editing work in tomato for characterizing the different stress tolerant genes and making the crop tolerant for specific stresses. So during this process, I have also published, as I earlier discussed, 7 different books in different publishers like Springer, Cabby, CRC Press. So these are my books. During this work, I have also published different chapters in different reputed publishers. So the earliest chapter was my PhD chapter, where I have discussed the genomics resources that are available for abiotic stress tolerance in Solanaceae family. Then the latest work, latest chapter is on the plant-specific bioinformatic tools that we can use for crop improvement, because mining and everything we need for different resources for designing the CRISPR or designing the marker and everything. So what currently I am doing, as I earlier discussed, I have got the SERB funded startup research grant, where my goal is to characterize the banana bunchit of virus for I have to characterize different banana germ plants for against the banana bunchit of virus through the transcriptomic approaches. So we have got different B genome of banana germ plant, where we are getting some tolerance level, and the work is now at the transcriptome level, where the transcriptome sequencing is going on, and we are hopeful that we will discover few genes based upon the comparison of tolerant and susceptible genotypes for that will give the tolerance and resilience traits. So institutional duties, currently I am scientist in charge for operation in commercial tissue culture lab. We have got DBT TCP team accredited commercial tissue culture lab at IGKV, where we are managing mass propagation of sugarcane and banana, and I am also in charge of phytosanitary laboratory. This is the APIDA certified GMO testing lab, where I am taking care of the GMO testing aspects of APIDA certified testing for different food products as well as crops. So these are my journey, and as you know, the science could not be done solitary. So I am very much thankful to the Society for Plant Research, the committee, the chairman, Vanna ma'am, for conferring me the SPR Fellow Award. I am very grateful for my foundational training under the Dr. Manoj Prasad sir. Because of that, I can do many things, and I am also grateful for NIPGR, Punanagar University, Amritsar, and Indira Gandhi Agriculture University for providing me the institutional backing, the infrastructure, and academic support. And with this, I will conclude my talk. Thank you very much.
Thank you, sir, for your excellent and informative presentation. Now, I take the privilege of inviting Dr. Vivekanand Mandal, sir, from University of Gaurbanga, West Bengal, to kindly deliver his lecture. Sir,
okay.
Over to you.
Okay. So thank you, and good afternoon to everyone. Today, I am here to just present the topic of this SPR Award lecture in front of you. First of all, on the onset of this, I express my sincere gratitude to Professor Bhatnagar, Professor Bandana Bose, and the distinguished member of the SPR committee for giving me the opportunity to deliver such a awarding me this SPR award 2026, and also giving me the opportunity to deliver a talk. So I am just sharing my presentation in front of you. Let me allow to share it. Okay. Yeah. Okay. So is it be visible? Is it
Yes, sir.
visible to you?
Yes,
Okay.
sir.
Okay. So yeah. So for this presentation, I chose one of my interesting or fascinating observations. That is on the allelopathic compounds from a particular plant. So before going in deeper side on this research, little bit of on this bioherbicide research, I want to just go through my little bit of bionote area. So regarding my academic profile, I did my graduation in botany honors from Vishwabharati University in 1998. I did my master's from there also with a specialization in plant physiology, biochemistry, and molecular biology in 2000. So later on, I qualified that CSR NET and also that CSR NET LS. And I did my PhD under the supervision of Professor Narayan Chandra Mandal and Professor Sukanta Kumar Sen from the Department of Botany, Vishwabharati University. Switching from that topic, from the plant physiology background to a microbiology background, and the topic of my research was on the production of bacteriocin by Pediococcus acidolactaceae for application as topical therapeutics and probiotics. So I also got some opportunity to get some ideas to work on our hands-on training on the microbiological area. So during the course of my service as a faculty member in different colleges and the university, I started my higher education teaching learning process in 2006 at the postgraduate department of botany in the Darjeeling Government College under that West Bengal Public Service Commission, PSC. Later on, I also got the regular transfer to that Hooghly Mohsin College, and then I joined to this University of Gaurbango in 2011 December. So since then, I'm here, and I'm in the capacity of professor of botany since 2014. Okay, so I also served that HOD as administrative capacity, and also served approximately 1.5 year as a capacity of the Dean of Science. And currently, I'm the project coordinator of WBD STBT Boost, funded by the Department of Science and Technology and Biotechnology, Government of West Bengal. This is basically a infrastructure grant received from the state funding agency to provide the basic laboratory facilities to the department. So by this time, during this course of my teaching learning process, I supervised 8 PhD scholars and 7 under supervision. I also supervised 2 MPhil scholars, and also there are 65 numbers of approximately master students who pursued their dissertation under my supervision. And today's topic, I will be presenting you. This is part of the one important this master dissertation work that insisted me to work on this allelopathic research area. So I also received one of the prestigious award, that is Best Researcher Awards in 2022. So my key research interest area is principally on that our antimicrobial compound discovery. It is from that our lactic acid bacteria, it is from that our fungal organisms, and also from that our medicinal plants that we are using ethnobotanically, and to decipher the active principles out of them to develop some novel antimicrobials for that disease mitigations, and also as well as for the crop protection and food preservation technologies. I also worked on supervised on some research on that our biopolymer degradation. As we know, the enzyme industry is very much is very much catchy. So we are trying to develop some or purify some these glycolytic enzymes like pectinases and the xylanases from the fungal organisms, and that could help in the solid waste management problem, as well as industrially relevant product development. And in addition to this, I also supervising some thesis on that plant microbe interactions, particularly on that our endophytes from that some hemiparasites, and also that our mycorrhizae. So this is the current status of my Google Scholar citation 1041 with the age index for 20 and item index 33. So I also conducted the project work, and three numbers completed, 4 numbers completed, and 3 ongoing. And I published 1 books, 1 edited book, and 1 book, 14 numbers of book chapter. I also received collaborative patents along with the chemistry from the Australian government. So since then, I also received the membership from the academic societies, like from that our Society from the Plant Research since 2026, and also Association of the Fungal Biologists since 2024, and the life member from that our this Association of Microbiologists of India 2007. So this is basically some brief bio note regarding my academics and some academic involvement in the research and guidance. So I want to just project this lecture in a just a little bit of some awareness type program with some focus on our research findings that we are working on that. You see, this is the basically the different photographs taken from that our Google databases. That this the agricultural field, which is very much prone to have different types of unwanted plant species, and they are basically are called the weeds. So the weeds and the weed mitigation is one of the leading challenge for the current agriculture practices. And most of the cases, we are just the industrialists, the pesticide industry, they are trying to develop more synthetic pesticides and that are weedicide, herbicide to control the shutdown. Still, many of these compounds are yet to have the good safety status. But due to the urgency, due to the need of our agriculture practices, we have to, or we are using the different types of these weedicides. So I want to, this is the basically the GMA's working field. This is the basically Parthenium, as you can see. It's very notorious, this weed. And this is basically Amaranthus, the different species of Amaranthus. They are growing in the agriculture field. So, and there is a news coverage in the Hindustan Times in 2018 that this weed invasion is causing the annual loss of this crop productivity, and the cost of such the loss is approximately $11 billion in India. So this is a big challenge. When I took this opportunity to work on this area, because we have to think for some developing some biopesticide, bioherbicide, so that the agricultural field as well as that enter this agricultural ecosystem remains quite in a healthier way. So out of this is one graphical presentation. You see that herbicides is yellow marked here, is approximately 47.5%. So it's quite high in comparison to insecticide, fungicide, and other pesticide that we are using today. So resulting in imbalances in the microbials and the ecosystems of the agriculture field. So if you go to this little bit of some literature perspectives, basically weedicides or the herbicides are those chemicals that are particularly used for killing or to suppress the growth of the unwanted plants in your targeted field. So whenever they are just occupying or sharing the space for the nutrition, for the water, for competing with their this canopy, all these are the big concern to have a good productivity out of that. Not only that, they are also deteriorating the quality of the seeds, because the seeds are having if it is the weed infested, then next generation cycle of the seed harvesting and the maintenance is little bit of difficult to have, because one wheat pan can generate 1,000 numbers of wheat seeds, and that is more difficult to handle all those things in the next generation cycle. So these are basically the overall the ideas of the pesticides. Out of these, this is glyphosate, one of the important the herbicide we are using today, and many of these organophosphorus, some of these are also being trial for that, are used for that our this weed control. Now, this is in perspective of that our 2023 that
Kindly, this is a request as the time it has slot has been allotted for each members. So
yes, I'm
the
moving,
time.
madam.
Yeah,
Okay.
okay,
I'm moving.
okay,
I'm
sir.
moving. Okay, I'll be faster. Yeah, definitely. So this is basically world scenario regarding how much we are using. These herbicides can be this pre-emergent, post-emergent, systemic herbicides, and also the contact herbicides. And this is all that our available herbicides that our farmers are using to control that mitigation. So the very common is glyphosate, the pendimethalin, and 2,4-D, and that pyribac sodium. These are all nowadays are popularly used. And many of these herbicides having a unique mode of action, targeting the different pathways. Now, if you look at the Indian scenario, that approximately 20 numbers out of the 87 herbicides are registered. 20 are popularly used by the Indian farmers, and many of these are having very high risk for our this health, as well as that our the residual problem in the agriculture field. So if we just look at these, that one of the leading this herbicide, that is glyphosate, now become resistant for that our weeds. So we have to think for the new this herbicide resistant crops, as well as develop some herbicides, and that is the issues I have to consider. That is the considering some biosafety align this herbicide that can help for the mitigation of this problem. Not only that, this is also having some residuals. So people are reading that pesticides are in our food and drinks, and glyphosate is also being noted as a carcinogenic for the human health. So I'm just moving to this perspective. You see, okay, and okay. So a journey towards the discovery of the allelopathic compounds, and that is one day. This is fascinating observation. We started from the gloriosa pot, and this is this. So you see, this is a Euphorbia tuberculi growing in our departmental space. And in the same plot, that one is the rhizome has been planted quite earlier, and that started germinating. So once this is germinating, what we have observed is this casulation acid, these CAM plants become drying day by day, resulting in ultimate destructions of the entire plant. So we assume that these gloriosa superba growing out of that mixed culture plot have some allelopathic potentials that caused the destruction of this type of highly resistant this CAM plant, that is Euphorbia tuberculi. So later on, we extracted the compounds from that the soil leachate, and as well as the rhizome extract of that gloriosa superba, and we prepared that extract with the name of that USL1 with the different combinations, so that they can be mitigated. So along with the kinetins, along with that our H2O2, and also, as we know, there's gloriosa rhizome is rich in colchicine. So yes, madam. So we did this type of experimentation. We also worked on some localization experimentation to do this thing, and also partially characterized, but yet to fully understand. So what we have observed that the USL treated seeds, we primed the seeds of Amaranthus viridis, and we observed that it's having germination inhibitory property, though the environment, the treatment conditions are the same, but it's failed to do. And also this kinetin, even the triggering factor of kinetin also failed to stimulate the growth behavior. So we prepared the 2 times, that is one, that is 6-hour priming, and as well as the 12-hour priming along with this treatment, and observed this sort of behavior after different course of observation. So you observed that there is a variations in the germination properties, soluble electrolytes, amino acid content, total carbohydrate, proteins, as well as the reducing sugar. And also this is the enzyme parameters like amylase, ascorbic acid, catalase, hydrogen peroxide, DNA content, peroxidase, as well as the RNA content. And you observed that there is a significant impairment of all these biochemical parameters in the treatment sets with the USL or that rhizome extract. So these are all that line graphs showing the decline of, this is 6-hour prime, this is 12-hour prime, and you observe this way. So this is also that statistical analysis. This is our PSA biplot, and also that our PSA biplot showing the groupings of the interaction phases. So these are all some data analysis perspective. Now regarding that our how this compound is exerting their effect to cause it, we did that ROS training on the different treatment parameters. What we have seen here.
Yes, just a minute. Professor
Yes,
Vivekananda.
Yes.
This is not a research webinar, you know, and we have to make it as a parameter, because out of 3 parameters for giving a fellow award of a best doctoral thesis, this has been kept as mandatory webinar so that you can showcase your achievements, and rather than giving the entire research findings. So it is consuming the time and it will go against the parameter. So kindly take a note of it. Thank you.
Okay, sir. I'm just concluding my idea, because I thought this topic could be relevant. That's why. Okay, sir. So this is a little bit of our understanding, and we also yet to fully characterize these molecules. But we are hopeful that this sort of behavior on that our targeted weed plant could be a future putative this bioherbicide. We published this work on this in a Vegetables journal. This is my little bit of research findings. I will also publish one edited book in the title Plant Microbe Interest under Xenobiotic Exposure. So these are my scholars who did this. These are the master students who worked on this allelopathic potential work. And I'm also thankful to the different funding agencies to giving me the funding facilities to the research work. And these are the research scholars who worked on in different perspectives. So thank you everyone for giving the opportunity.
Thank you, Professor Vivekananda.
Thank you, sir,
presentation.
for your informative lecture.
Actually, yes, Jyoti.
Yes,
This is my humble request, because during some other time we shall be having a detailed academic deliberations from all our esteemed awardees, because you know that we are facing a big constraint of time, and this is one very important requirement for the award of fellow or for other best doctoral thesis awards. So as a parameter, we have kept this webinar. So we will certainly take advantage of your expertise and of your researches, and a separate webinar shall be arranged accordingly on the basis of the expertise of our awardees and other scientists, invited scientists from different countries. So it is my humble request that you kindly focus more on the academic journey within 10 minutes time, and within that time, the complete showcase of your career during this time, which has been considered for conferring this award, may kindly be highlighted. So I'm sorry to intervene between the lectures, but I thought that this is very important, and because we want to take advantage of your researches, and everybody who are sitting over here, they would love to listen to the research component, but we will arrange some dedicated seminars sometimes later. But today it is a requirement for the SPR award. Therefore, we have to refrain going beyond 10 minutes time. Thank you all very much. Thank you. Dr. Jyoti, please continue.
yes, sir. Okay, sir. Now it's my honor to invite Dr. Prashant Ji Kanwar, sir, from Directorate of Floriculture Research, Pune, to kindly deliver his lecture. Sir, kindly proceed with your presentation.
Full-fledged करायचं का असंच पुरेसं आहे? Yeah. Thank you, Dr. Jyoti. I would like to thank Dr. Professor S.K. Bhatnagar, Dr. Vandana Bose, and Dr. Dolly Vataldhar, Dr. J. Narayanan, and Dr. Jyoti also for giving me this opportunity. Am I audible to you?
Yes, sir. Yes, sir. You are audible. And sir, timer is also going on the screen.
Okay, ma'am. No issue.
I think it should be visible to you also.
Okay, ma'am.
Yes, sir.
Yeah, thank you very much. This is not moving.
No, sir. Slides are not moving till yet.
Slides are not moving.
No, sir.
Yeah, just wait for a second. Sharing cancel. तिथून आता हे कर पहिले फुल करून फुल स्क्रीन कर
Dr. Prashant, you have not sent a PPT to our office?
I haven't shared, sir. I will share after this immediately.
No, that was required only because sometimes there is a technical glitch. So in
Yes.
order to overcome, our office can run the slides. That's why it was asked. But perhaps it
Actually,
was not sent
I
anyway.
was out of station, sir. So that's
Okay.
because of that it was a
Okay.
problem.
Please continue.
It's not moving. That is the problem only.
Sir, if possible, meanwhile, you can share the presentation with the office.
That will be better. You kindly share it and we will take up just after the next presentation.
Okay, sir. Okay, sir. You can proceed with the
please
next
share
one.
it.
I will.
Dr. Jyoti, please
Okay.
invite
Yes, sir.
Dr. Umesh Thapa.
Okay, sir. So, may I now request Dr. Umesh Thapa, sir, from Bidhan Chandra Krishi Vishwavidyalaya, West Bengal, to kindly deliver his lecture. Sir.
Yes, ma'am. Just a minute. I'm sharing my slide, ma'am.
Okay, sir.
Just a
Okay.
minute. Is it visible, my slide, ma'am? Hello, my slide is
Sir.
visible.
No, sir. I think not yet.
Ma'am, you have my slide, so you can, because I'm not like getting to, you have my slide, so can you show
Sir.
me?
Okay, that will be done from the
Okay,
office.
ma'am. Okay, now is it?
No, but
No.
forget about. Office will do.
Now, is it okay, sir?
Yeah, it is okay.
It is visible, sir.
okay? Yeah, okay. But it is again not moving. Same thing has happened over here also.
Sir, if you are having a presentation with us, if you have shared, then we can do it for you.
Yes, ma'am, you have. Then I stop my sharing. You just show me your presentation, ma'am. I'll start from there. I've
Okay,
given
sir.
you the presentation.
Okay, sir. We'll request to the office. Yes, sir.
Okay, good afternoon to everybody. Myself is Dr. Umesh Thapa. So in a brief, before giving this presentation, first of all, I would like to thank Dr. Shubhat Bhattanagar, Madam Bosch, and all esteemed organizations of this Plant Research Society. It's my pleasure to be here to honor the fellow of this society. Thank you very much to all of them. So today I want to show the achievement on cost-effective soilless farming for the high-value vegetables. That is our new avenue for the urban entrepreneurship. Next slide, ma'am. Next slide, please. So before that, I just want to highlight it as because why I'm choosing this subject as my research achievement, because nowadays in the present scenario, the hydroponic becomes very much popular for the urban and the peri-urban areas. As we all of you know, like as because of these difficulties of the agriculture sectors for the farming community, with the urbanizations, with the industrializations, with the climate changing, the people are suffering from the cultivation of the good crop to get the highest yield. So for that, I chosen this topic as well as I chosen this subject for my achievement, because these works can be fitted very well in case of the urban and the peri-urban areas, so that we can grow our vegetables in our flat cultures, in our urban areas, in the peri-urban areas, so that we can get the fresh vegetables. As we know, most of the vegetables which we are consuming are getting from the farmers. They are all full of the toxicity. So to overcome this problem, this research work, I have taken my challenges. So the modern hydroponics begin with the researchers like the Dr. William Frederick, but it is not a very new topics for the hydroponics. As we see about the previous time of the hydroponic, in the history of the hydroponic, the floating garden of the Aztec of the Mexico and the hanging garden of the Babylon shows that the hydroponic was there since from the long time. If we think about the Indian scenario, basically I am from the district of the Darjeeling, and the district of the Kalimpong was the first area which has been introduced for the hydroponic in the year 1946. This way, the hydroponic has been popularized in our country also, but due to some of the reason, the hydroponic does not become very much popular in that season. But now at the present moment, in the 20th centuries, everybody was jumped about to the hydroponic and the soilless technology. So for that reason, I want to focus the highlighted of my achievement in this STEAM organization. Next slide, please. So here what I'm talking about the low-cost hydroponic technology for the high-value vegetable crops. Again, why I have chosen the high-value vegetable crops? As you know, as because of the present scenario of the food habit changes of the young generations, people like, they are very much fond of having the foods like the McDonald's, Pizzas, Hot, Domino's, KFC, Subway, etc. So for this, the hydroponic, the high-value vegetables will be found to be very much suitable, because almost all these high value vegetable crops like the red cabbage, broccoli, colored bell pepper, lettuce, asparagus, etc. is one of the vegetables which can fit it to have such type of the food habit, because these vegetables require preparation for that pizza, Domino's, whatever may be. Because nowadays at the present scenario, the young generation doesn't want to consume the chapatis, tarkas and like that. They are very much fond of having the chowmin and like that. So for that, these vegetables can found out the suitable places, because the combination of the vegetables will be very much required for this high value crops. That is why diversification of the high value vegetable crops in our country through the soilless farming and the hydroponic is very much essential. As you know, most of the exotic vegetables or these high value vegetable crops are grown in the city market, and nowadays each and everyone are jumped about to consume these vegetables. Even we have seen in the cities, in the mall, in the big cities, in the five-star hotel, in the multinational fast food chains, all these high-value vegetable crops suited the best places. So for that reason, I have chosen for my achievement by doing the research work for the high-value vegetable crops in spite of the traditional vegetable crops through the soilless farming as well as the hydroponic technology. Next, madam, please. So for these vegetables, for the hydroponic or the soilless technology, these are some of the media, because as we know, in case of the hydroponic and the soilless technology, we cannot grow the vegetables in the soil. That is a soilless free. Either we can use a substrate as a water, or either we can use a substrate, organic substrate like the sawdust, cocoa peat, wood chips, etc. Otherwise bark, others even in our research, we have found out we have standardized the substrate of the vegetable kitchen waste also. So these are some of the media which we cut for to raise the hydroponically or the soilless farming system of the vegetable crops. Next, please. So this is what I want to highlight, the photographs of my research activities. This is a cost-effective hydroponic soilless farming for the high-value vegetable crops. So in these systems, we can grow almost all types of the high-value vegetable crops. Here in the slides, it has been seen the first hydroponic setup, which I have purchased after completion of my postdoctoral research from the University of Turin in the soilless farming. I came back in my university 2010, and I purchased this setup. And this setup consists of 42 plants, cost about that time in 2010 more than the 14,000. So for that, in our university, we himself developed our very low cost technology of the PVC pipe made. This one, the very low cost PVC pipe made NFT system. This is considered NFT system. So it is now at this present moment also we have developed, and it is very much popular to the urban areas and the people who are living nearby the cities in the flat and like that. They have a very high demand of this PVC NFT system, which cost about only 3,000 10,000 rupees. So in this system, as a vertical as well as a horizontals, we have access so many vegetable crops, including almost all the high value vegetable crops, including lettuce, spinach, French bean, onion, broccoli, sprouts, Brussels sprout, pumpkins, everything. So this is what we have developed, one of the very low cost NFT system in our university with my leadership. Thank you. Next slide, please. So now this is one of the another cost-effective hydroponic production system which we have developed using the recycled mineral waters. This a cost-effective hydroponic production system which has been used by recycled mineral water bottles has been developed for the cultivation of the leafy vegetables, which has to be in a very under limited space condition. So this technology utilizes the discarded plastic mineral water bottles as growing containers, thereby converting the waste materials into the productive units for the sustainable soilless vegetable production. So this system was found very highly suitable for raising. This is one of the example of the vegetables like the palang sak, Beta vulgaris, bengalensis, which you have raised. We have standardized the media. Here we have chosen the medias like the hydroponic purely by the water, purely by the waters, and some of the substrate with the coco peat versus coco peat with the mixtures of the perlite and as well as the vermiculite. And another substrate just now which we discussed about, that we have developed the vegetable kitchen vegetable waste. So we have composed the kitchen vegetable waste substrate, and that substrate can also be utilized. That means the vegetable kitchen waste, peel of onion, peel of potato, everything. We have classified the cucurbitaceous vegetable crops wastage, leguminous vegetable crops wastage, solanaceous vegetable crop wastage, so that we have found a very suitable substrate by using this vegetable wastage in the very low cost effective hydroponic system by using this recycled mineral water bottles. Next, ma'am, please. So this is another one. This one, another one which we consider as a low-cost floating system of the hydroponic system. As you know, in the floating system also becomes the very much popular in our country. So here, what we did in the low-cost hydroponic system, very low-cost floating hydroponic system was developed using the thermocol seed, which you have seen over there. These are the thermocol seeds as floating place over the nutrition solution filled with the trays. Here we are filled in the tank, and here we are filled with the trays. Okay. Holes were made at the proper spacing to hold net pots, and recycled plastic cups were used at the low-cost net pot. Here we are seeing the net pot, but in the latter stages of research, we have used the low-cost wastage of the plastic cup also. So this system is also found to be very suitable for cultivation of the spinach, lettuce, even the French bean also. The French bean also comes up very well in this technology. So this technology which we have developed is a very economical, water efficient, portable, and easy to manage, and ideal for now in the present scenario of the urban and the peri-urban garden for the rooftop terraces and the urban vegetable production system. This system becomes the very, very beautiful system in the very low-cost technology. Next, ma'am. Next, and this is another one which is considered as a low-cost Dodge bucket hydroponic system, which we have also developed by using the recycled plastic container. So this is low-cost Dodge bucket hydroponic system also developed by using the recycled bottles. Like, see, this is the bottles of the sweets or the containers of the biscuit, and this is a buckets, low-cost bucket by throw out of the painting buckets. So this can also be utilized to raise this system in the soilless of the hydroponic system, which we develop as a low-cost Dodge bucket hydroponic system. Next, please. So this is one technology which we have developed in the capsicum cultivation. Here we have ashes, the different colors of the capsicum, red, yellow, and the green, and with the different set nets. Like as we generally in agriculture and horticulture sectors, we have seen that most of the green set has been utilized, but we have assessed with the green set with the different percentage, 50, 35, and the 75% is white set net, red set net, and the black set net in capsicum cultivation under the soilless as a back system. We have developed that this white set net found to be very much ideal for the development of the color and encashment in the soilless system as a grow back system. Here we have standardized the ratio of the 2:1:1 of soil substance media like the coco peat, vegetable wastage, and vermiculite. So this is one of the technology which we have developed. Next, ma'am. Next, and this is what I just want to highlight it.
Sorry to interrupt, sir. The time sir has been
Just
completed.
2 minutes, ma'am. So
Yes,
this
sir.
is a low-cost portable PVC frame structure which can be very much easily constructed, and that can be dismantled very easily after completion of the work. As we know that in the low-cost, bamboo is considered as a very low-cost technology, but bamboo is priced about 250 rupees a single. So that this technology also found to be very much ideal. Next, ma'am. Next, and the last one, this is a cost-effective biodegradable proctor system. We know by raising the seedlings of the vegetable crops, first of all, we used to raise in the seed bed, but as because of the high-tech technology, we have adopted the proctor technology. But now, in spite of the proctor, we have developed this type of the banana leaves by using the banana leaves as a cup shape, and within that banana leaves, we put the cocoa peat, and here we directly put the seeds, and that seedlings has been transferred to the main field without uprooting. Same, and because this banana leaves is a biodegradable, with the whole biodegradable leaves of the banana as a crop or the crop tray, we transplanting the plant in the field. This is also one of the cost-effective biodegradable crop tray using banana leaves we have developed. Next, ma'am. This is a microgreen that we have also worked about, and we are doing the works on the microgreen. So we have standardized the different intensity of the light. In the photographs, it has seen with the different colors of the light. Now we have decided and we have found that the white light found to be very much ideal for the microgreen. And nowadays the microgreen is considered as one of the very nutritive and high antioxidant vegetable crops, which can be good for the health, and as well as in the fodder rice. This is the crops of the rice bean. So this rice bean fodder microgreen can also be raised very quickly in the hydroponic system, which we have developed, and this system is found to be very much ideal as a microgreen consideration. Next. And this is the last one, I think. So this is a thanks. And now at last, I'm Dr. Umesh Thapa. Now at present, I'm working as a professor in the Department of the Vegetable Science, Bidhan Chandra Krishi Vishwavidyalaya. In the academic, I have so many experience of the administrative of different posts, direct extension education, head of the department, placement chairman, and DSW. And I have so many research work I'm handling. And that is what in very short. Thank you very much. And thank you very much. 1 second.
So thank you, sir. Thank you so much for your insightful lecture and valuable scientific perspective. Now I would like to invite Dr. Prashant Kavar, sir, to kindly deliver his lecture. Sir, you can proceed with your presentation.
Dr. Prashant, kindly continue.
Very good. Yeah, just sir. I'm just starting, sir. Yeah, at the outright, good afternoon to all of you. I thank Dr. Professor S.K. Bhatnagar, sir, Dr. Hanchinal, sir, Dr. Vandana Bose, and Dr. Dolly Dhar, Dr. Jay Narayan, for considering me for this award. I thank the society as well. Yeah, this is my biographic about this. I did my master's in agriculture biotechnology from Indira Gandhi Agriculture University, Raipur. Then I joined at Vasantdada Sugar Institute as a scientist. After this MSc, then in service I did PhD from Shivaji University, Kolhapur. During this my scientific career, I got DBT Chris Fellowship to work at John Innes Center, UK, Norwich. I got National Merit Scholarship, then ARS NET. Then I was organizing committee member of 8th World Congress at Netherlands. And recently I got the Genome Editing Fellowship from ICAR to work at Texas Tech University. I have total 51 research publications, 3 books, some book chapters, then 1 variety, then biological documents, then TV talks, and other publications. I have 58. Almost I have 1200 plus 1200 plus database submissions, and now 4 whole genome sequence submissions. Basically, I'm working here at DFR since 2015 as a senior scientist, and this is the only institute in Asia, we can say like that, dedicated for flower and ornamental crops. So our mandate is to work on flowers and ornamental crops. Initially, I have given my efforts in establishment of this institute, but this is a national institute for floriculture crops and ornamental crops. I'm supposed to work on biotechnological aspects, all the aspects of this institute. So initially we worked on development of unique DNA fingerprints for flower crops. As you know that flower varieties, when they change the hands, the names are changed, and many conflicts of interest are coming in between. So we are using SRAP, ISSR, then SCOT system for developing DNA unique fingerprints of flower crops. Almost we have developed for gladiolus, tuberose, heliconias, chrysanthemum, and roses. The germplasm available DFR. Then we are doing the pollen morphological studies for all the crops also in collaboration with Shivaji University, Kolhapur. In this also, we have almost developed pollen morphological traits for gladiolus, rose, and chrysanthemum mostly, like 148 accessions of chrysanthemum, 75 for gladiolus, and almost 120 for roses. So this is the work done in collaboration with Shivaji University, Kolhapur. These are the glimpses I'm giving about my research. Then we worked on harnessing the natural pigments, nutraceutical pigments from chrysanthemum, rose, and marigold. Then we are working on aromatic organic VOCs and gene expression studies of ornamental crops. In that, we have completed rose cultivar studies related to scent analysis and tuberose. Then we are developing some bioactive compound extraction processes at ICAR DFR. I will just scan through the things. Like this is the chrysanthemum cultivars we have studied. We have published this paper in Genetic Resources and Crop Evolution. We have completed for almost chrysanthemum 22 chrysanthemum varieties. In case of rose, we have completed 17 rose varieties. Then in case of marigold, 22 varieties, we have did all the studies for the compounds which are available in marigold flowers. So this is the glimpses for that. Then we did the VOC analysis for fragrant and non-fragrant rose cultivars, and we have published this paper in Physiology and Molecular Biology of Plant recently in 2024. Then we did similar kind of studies for single and double type tuberoses in tuberose, and we could identify specific scent related compounds in single, as singles are mostly scented and doubles are less scented. So what are the compounds, what are the genes, how the gene network work, that also we have done. This is recently in 2026, in this month only it is published in the Plant Molecular Biology, published by Willy. So this is the like gene network studies and all those things we have completed. Then we have developed, like my major area of working is metabolite engineering, and I have a big project for the same from ICR in genome editing initiative. So I have to develop a method for extraction, like green extraction of lutein from marigold flowers. So we applied three different systems, like ultrasonication based and microwave based and conventional based, and that we have studied. This is also, this publication is already in review in Food Chemistry X, and we found that that ultrasonication based assisted extraction method is very effective, because this is very commercially important. Lutein is very commercially important ingredient for pharmaceutical as well as nutraceutical industries, and we have developed ultrasonication assisted extraction method for marigold at our institute. Then we are using some, we are working on abiotic stress management in ornamental crops also. We are using integrated approaches for reliable screening and identification of salt tolerant chrysanthemum types, and silicon mediated strategies for enhancing salt stress tolerance in chrysanthemum. So this studies also we have completed, and we have developed a method wherein we can identify the salt tolerant genotypes, so that they can be taken up for breeding program. So this methodology is already established. This is also in review stage in one journal. So we did all kind of studies. In this case, this is a silicon mediated strategies for enhancing salt stress. We have utilized silicon as well as silicon nanoparticle developed at CUH, Haryana, for mitigating the salt stress tolerance. This study also we have completed, and this publication is in plant nanobiology. The main area of my work is metabolite engineering, which I have got DPTK's fellowship to work with Kathy Martin and John Innes Center. There, I have worked on two aspects, like use of AT-MeP12 transcription factor to enhance the novel phenylpropanoids. She has already worked on purple tomatoes, and similar kind of strategy we have used to increase the resveratrol content, which is only present in grapes, but we could develop such a technology there by using two different means transcription factor and the gene which is STSY gene, which is from Vitis vinifera, and that combined expression of these genes have given us like 100-fold higher levels of resveratrol in grapes, in tomato as compared to grapes. And second is isoflavone rich tomato also we have developed, like genistein, which is very useful pharmaceutical compound for breast cancer in generally in women. And in that also we developed, we used like overexpression of IFS gene and overexpression of AT-MeP12, and one background knockout for a DFR gene has yielded like 100 times more genistein in tomato, which is not the compound of tomato as such. And this work we published in Nature Communication in 2015. And now she has already commercialized these products in US and UK. That company is there. She is floated one company, and it is available for public, although it is a GM. Then similar lines, I'm working here in DFR, like identification of novel genes which can be modulated by genetic engineering or by genome editing. So I'm trying to find out genes for especially for the scent related compounds which are responsible for developing a variety specific scent in roses as well as in tuberose. And then of work is lutein enhancement and zeaxanthin enhancement and development of applied inducer lines in marigold, and then day neutral chrysanthemum using genome editing. As such, we see that genome so these are the studies we have completed already, and these are publications in review. Like we have identified the variety specific scent related genes in roses as well as in tuberose. Then we are doing that. We are doing genetic engineering also. Like we are using Virki 35 transcription factor from Arabidopsis, overexpressing it into the marigold, so that the flux pathway can be enhanced for the production of lutein or zeaxanthin. So we are developing transgenic in under the control of CCD4 promoter, which is a flower specific promoter. So we are doing transgenic marigold also, and we have successfully have some lines already with us. This is the media composition for regeneration and transformation. We are standardized. This is the actual optimized protocol for direct organogenesis in marigold variety Pusa Basanti Genda. And this is the pathway wherein I'm doing genome editing, so that LCYB, if we knock out, the flux will move to lutein production. This is the basic concept, and we are working on this. We have already developed vectors for genome editing, and presently we have almost 45 plants, PCR positive plants for Cas9, and lines we are proceeding for amplicon-based sequencing. So once they are done, then we will do
Sir,
the studies.
sir,
Okay.
sorry to interrupt, sir. The time limit has been
Yeah, I'm about
completed.
to
So
finish.
please.
Yeah.
Okay, sir.
So we are having this many lines already for LCYE for zeaxanthin enhancement. Then this is like transgenic work I told. We have already some lines for that. Then we are developing applied induction system for marigold, so that in breeding cycles we can have easy method to develop marigold hybrids be commercially viable. So for that we are targeting the DMP 8 and 9 genes in single and multiplex way, and there also we have some plants already with us. In case of chrysanthemum, as we know that chrysanthemum is a short day plant, so only flowering we occur in short day conditions, and I'm trying to modulate that it will give more flux. This is the chemistry or science behind that, which is already proven by Japanese people, and I'm using these anti-fluoresceins to knock down by using this thing. Already we have developed a protocol for chrysanthemum also, and we have already almost 30-35 plants which are positive for Cas9, and we will be doing application sequencing for them. Other than this, we are working on genome sequencing and transcriptome of ornamental plants. Already we have sequenced 3 marigold genomes, and with the analysis is on the way. We have sequenced one Bakul species, this Minipus elangi, by pack biosequencing. So we are contributing in that area also. These are the analysis we did for whole genome sequences of those. Then we are doing some bioinformatic work also to identify some important genes in important species. I have been given fellowship to go for US for 3 months to develop tissue culture free genome editing in ornamental plants. I have successfully developed the protocol there, and I have got some plants. I have constructed the vectors for all the genes which I am working on at present, and these genes are in different stages of culture. The plants for this are in different stages of cultures. It is very easy method. If you allow me, I can show that video, but I don't think it is working yet. So this is work already done, and this is a patented technology by Texas Tech University. Same I am applying here for marigold, chrysanthemum, and gerbera at my place. These are the vector constructs I have developed, and I have started working on gerbera also for powdery mildew resistance at ICR DFR. This is at initial stage only, so we don't have. These are my collaborators. I thank my collaborators as well as the associated scientists and students who are working with me. These are some PhD students and my RASRFs in my projects. I acknowledge and thanks them. I acknowledge the director, ICR Directorate of Floriculture Research, Director ICR Central Potato Research Institute, then Director General Vasundhara Sugar Institute, Pune, then Director Indian Agriculture Research Institute, Pusa, New Delhi, ICAR and SPR Fellowship Committee, and all the members, and especially Dr. S.K. Bhatnagar, Dr. Vandana Bose, Dr. Dolly, Dr. Narayanan, Dr. Jyoti. Thank you. Thank you very much.
Thank you, sir. Thank you for your presentation. Now, I would like to invite, it's my pleasure to invite Dr. Myrene Rosalind D'Souza, ma'am, from Mount Carmel College, Autonomous Karnataka, to
Yes,
kindly deliver her lecture. Ma'am, we warmly welcome you. You can start with your presentation. And ma'am,
sure.
it's a humble request.
Hmm.
Kindly complete your presentation within the time limit.
Sure, ma'am. I just share the screen.
Okay.
Is the screen visible now, ma'am?
Ma'am, till now, I think it's not visible.
Okay. 1 second. Now I think it should be visible, right?
Mm. Ma'am, have you started the slide share, sharing the slides with us?
Hello, I think I just got logged off, ma'am. Can you share the screen?
Okay, ma'am.
Okay.
Have you shared your presentation, ma'am?
Yes, yes, I have shared it.
Okay, ma'am. Okay.
Okay.
Okay, ma'am. I'm requesting office to kindly share your
Sure,
presentation.
sure, ma'am. Yeah, I'll just begin. Yes. Okay. Good afternoon to everyone. I'm Dr. Myrene Rosen D'Souza, HOD and Associate Professor in the Department of Biochemistry, Mount Carmel College. My study focuses on exploring a plant-based nanotechnological strategy for combating neurodegenerative disorders, particularly Alzheimer's disease. The work integrates phytochemistry, nanotechnology, zebrafish behavioral neuroscience, biochemical analysis, histopathology, and a bit of computational biology. The central aim of this research was to investigate whether green synthesized silver nanoparticles prepared from moringa leaf extract could provide enhanced neuroprotective effects compared to the crude plant extract. Next slide, ma'am. Alzheimer's disease, as we know, is characterized by progressive and irreversible neuronal degeneration that ultimately leads to memory loss, cognitive decline, and impaired reasoning and behavioral disturbances. The disease pathology is largely because of amyloid beta plaques and intercellular neurofibrillary tangles. The plaques interfere with neuronal signaling while the tangles disrupt intracellular transport systems. Together, both these changes lead to neuronal dysfunction and eventually cell death. In this study, what we tried to do was we used aluminum chloride to induce neurotoxicity as it mimics several pathological features seen in Alzheimer's. It alters blood-brain barrier permeability, disrupts cholinergic neurotransmission, and also promotes severe oxidative stress through lipid peroxidation. Although there are several drugs available in the market for Alzheimer's, these are mostly providing only symptomatic and temporary relief and do not reverse disease progression. So therefore, there's an urgent need for safer multi-targeted neuroprotective agents which are able to reduce oxidative stress. So our main aim was try to reduce oxidative stress and then restore neurotransmitter balance, and also maybe at a larger stage or longer stage in protecting neuronal architecture. Next slide, ma'am. Moringa, we know, is commonly called as a miracle tree because it has immense medicinal and nutritional value. The plant is rich in bioactive compounds, particularly quercetin, campherol, phenols, flavonoids, and many other antioxidants. Now, these phytochemicals possess strong free radical scavenging activity, and this is what we are trying to utilize here for neuroprotection. The crude plant extract suffers from various disadvantages such as poor bioavailability and limited blood-brain barrier penetration. So to overcome this limitation, we try to employ green nanotechnology. So in green synthesis, the plant phytochemicals will serve both as reducing agents and stabilizing or capping agents during the nanoparticle formation. Silver nanoparticles synthesized using plant extracts will be considered biocompatible and hence environmentally friendly. The major advantage of using moringa silver nanoparticle would be its high surface to volume ratio, enhanced cellular interaction, improved stability, and potentially increases penetration across the blood-brain barrier. So our hypothesis here was that moringa silver nanoparticles could exhibit increased penetration across the blood-brain barrier. Coming next slide, ma'am. The methodology for extraction and nanosynthesis. In the first phase of the study, we used the leaves of moringa, which were collected, dried, powdered, and then subjected to oxalate extraction. For green synthesis, the extract was reacted with 1 millimolar silver nitrate solution, The phytochemicals present in the extract reduce silver ions into silver nanoparticles. Formation of nanoparticles was visually confirmed by a color change due to surface plasmon resonance. The synthesized nanoparticles were then purified using centrifugation at 6,000 rpm. And after purification, phytochemical characterization was performed using UV-vis spectroscopy to monitor the nanoparticle formation, X-ray diffraction to study crystalline structure, FTIR spectroscopy to identify functional groups involved in reduction and capping, and SEM analysis to give us information about the morphology and surface structure. In addition, computational studies were carried out using quercetin and camphorol as target ligands. Docking studies, ADMET analysis, string network analysis, and Keck pathway enrichment were also conducted to understand molecular mechanisms and pharmacological interactions. Next slide, ma'am. The zebrafish experimental design is as follows. First, we acclimatize the fish under controlled lab conditions before experimentation. The animals were divided into 4 groups. The first group served as a normal control. The second group received aluminum chloride to induce neurodegeneration. The third group received moringa extract treatment, whereas the fourth group received the moringa silver nanoparticle treatment. Behavioral analysis was conducted using behavioral tracking assays. Following these studies, brain tissues were collected and homogenized. Biochemical markers such as acetylcholinesterase, malondialdehyde, and glutathione were estimated. Finally, histopathological examination was carried out using hematoxylin and eosin staining to observe structural neuronal damage as well as recovery. Next slide, ma'am. Next slide. So quickly going on to the results and discussion, both qualitative and quantitative analysis was performed. This is just the introduction. It was seen that when we looked at the qualitative analysis, alkaloids, flavonoids, terpenoids, phenols, and carbohydrates were present, which is normal in such a medicinally important plant. Then going on to quantitative analysis, we found that the total phenolic content was approximately 71 mg per gram. Total flavonoids was about 2.8 mg per gram, and the total tannin content was 17 mg per gram. These compounds we know will play a very important role in nanoparticle synthesis. Phenolic compounds donate electrons rapidly to reduce silver ions, while flavonoids stabilize nanoparticles by acting as natural capping agents. Therefore, the phytochemical richness of moringa not only contributes to antioxidant activity, but can also help in successful nanoparticle synthesis. Next slide, ma'am. Coming to the characterization of silver nanoparticles, UV visible spectroscopy. I think next slide, ma'am. In slide on characterization. Ma'am, you're on the first slide.
Dr. Myrene, you were supposed to give your entire academic status, what had been your achievement during your career,
Yes,
which
sir.
has been considered for fellow award. It was not required. Presentation of your complete research paper or
Okay,
research
sir.
article was not required. We were
Yes, sir.
interested in your entire academic career, which was taken as a criteria for
Okay, sir.
your selection as a fellow of SPR. Rather, it has been, I don't know whether it has been overlooked or you could not see the mail. Anyway,
Yes,
conclude it
sir.
now.
I thought it was a paper that we had to present, sir.
No. Anyway, please continue.
Okay. Okay, then moving on to characterization of the silver nanoparticles. In UV-vis spectroscopy, we see that a characteristic absorption peak is observed around 427 nanometers, which is a typical indicator of silver nanoparticle formation due to surface plasmon resonance. FTIR analysis revealed important functional groups involved in nanoparticle stabilization. 3 peaks were noted, which corresponded to stretching associated with phenolic compounds. Methoxy and alkyl groups as well as carbonyl functional groups were associated with these peaks. SEM analysis demonstrated, next slide, ma'am. SEM analysis demonstrated that the nanoparticles were predominantly spherical and uniformly distributed, whereas EDS analysis confirmed elemental presence of silver. XRD analysis validated the crystalline nature and face-centered cubic structure of the nanoparticles. Together, these characterization results confirmed the successful synthesis of stable and biocompatible moringa-based silver nanoparticles. Next slide. Moving on to the toxicity testing and locomotor activity. Based on preliminary testing, 100 mg per liter of plant extract and 0.2 microliters per milliliter of silver nanoparticles were selected for the treatment. The locomotor activity test was performed to assess neurological function. Aluminum chloride-induced zebrafish exhibited significantly reduced movement and shorter travel distances compared to the control group, indicating impaired neurological coordination and anxiety-like behavior. Treatment with moringa extract improved locomotor performance. However, the silver nanoparticle-treated zebrafish showed even greater recovery. In speed analysis, the control fish moved at approximately 62 millimeters per second, whereas aluminum chloride-induced fish moved at only 38 millimeters per second. Moringa treatment improved the speed to approximately 52 millimeters per second, whereas moringa silver nanoparticle treatment further improved movement speed to around 57 millimeters per second, indicating that nanoparticle treatment produced superior behavioral recovery. Next slide, ma'am. Coming to the neurobehavioral analysis, additional to this, we had in order to evaluate anxiety, learning, memory, and exploratory behavior. The first test that was performed was a novel tank diving test, which measures anxiety-related behavior in zebrafish. So normally, a healthy fish will gradually explore the upper regions of the tank, as seen in the picture, whereas aluminum chloride-induced fish will show reduced exploration and increased bottom-dwelling behavior, indicating anxiety and neurological stress. Both moringa as well as moringa produced silver nanoparticles treated fish demonstrated improved exploration patterns, as can be seen in the fourth panel of the first slide. In the spatial recognition test assessed, which helps to assess memory and cognitive performance, it was seen that the treated groups displayed better spatial recognition compared to induced group. Inhibitory avoidance tests evaluated learning as well as memory retention. So nanoparticle treated zebrafish exhibited near normal behavioral responses and improved memory retention. Overall, the results of all these three tests suggest that moringa silver nanoparticles possess significant neuroprotective potential compared to reversing aluminum chloride-induced behavioral defects. Next slide, ma'am. This was the biochemical evaluation. Acetylcholinesterase, we know, is responsible for breaking down.
Ma'am,
Yes,
sorry
ma'am.
to interrupt.
Yes, ma'am.
The allotted time has been completed, ma'am. So if you kindly wind up with it.
Wind up. Yes, ma'am. Okay.
Okay, ma'am.
Yes. So treatment with moringa improved all the three mentioned parameters, which is acetylcholinesterase, malondialdehyde, as well as glutathione. And the moringa treated silver nanoparticle treated group was found to demonstrate superior restoration of biochemical balance. Next slide, ma'am. Okay, same slide, previous slide. This is the histopathological assessment. Previous slide, ma'am. Treatment with hematoxin and eosin staining, we saw that the control group exhibited normal neuronal architecture. In contrast, aluminum chloride treated group displayed neurological degeneration, pycnotic nuclei vacuolization, cellular shrinkage, as well as disrupted brain architecture. The moringa treated group showed partial restoration of all these parameters, which I just discussed, whereas the silver nanoparticle treated group demonstrated remarkable improvement in terms of neuronal cell appeared healthier, tissue integrity was much better, as well as degenerative changes were significantly reduced. Next slide, ma'am. So here we see the molecular docking and ADMET analysis. Docking analysis demonstrated strong binding affinity of both ligands mentioned towards target proteins associated with Alzheimer's disease. The interaction suggests potential inhibition of amyloidogenic pathways and possible neuroprotective activity. ADMET analysis showed both quercetin and camphorol penetration of blood-brain barrier was also seen. So next slide. To conclude, overall nanoparticle formulation demonstrates superior neuroprotective efficacy compared to the crude extract. Therefore, the study highlights therapeutic potential integrating phytochemistry with green nanotechnology for future degenerative disease management. Thank you, ma'am.
Okay, thank you so much, ma'am, for your
Yes.
presentation.
Thank you,
And
ma'am.
now I would like to invite Dr. Shufia Khan, ma'am, from Dr. Bansidhar Institute, Gurugram. Ma'am, kindly deliver your lecture, and the platform is now yours. You can start with the presentation. Dr. Shufia Khan, ma'am, are you there? Sir, I think ma'am is not in the list.
Dr. Sufia should be here. She is online.
Dr. Shufia, ma'am?
Hello.
Yes, ma'am. You're audible. Ma'am, you can start with your presentation, ma'am.
Yeah, yes. So may I share my presentation?
Yes, ma'am. Yes, ma'am.
Yes. So is it like visible?
Yes, ma'am. It's visible.
Yeah. So good afternoon, all the respected dignitaries, Professor Bhatnagar, or committee members, and the distinguished guests. It is a great honor to deliver the today's SPR Fellow Award lecture. And today I will take you through my own journey from the last 20 years, like I was working in the plant biotechnology, molecular biology, and the water and the wastewater recycling technology. And when 2 decades back, I didn't think about the about the patents, translation, entrepreneurship, and the startups. But over the years, I have realized that that research publication is not the end of the journey. So a scientific discovery must create the impacts. So I will take you through my own journey that can easily depict you that how the field or the basic research can convert it into the field deployment for laboratory experiments to the industry pilots, and from academic publication to the entrepreneurship or establishment of the startups. So my talk will be on the arts and science of the translation, where I'm bridging the science industry and the society. Sorry. So this is the like the various institutions have shaped my career. Today, currently, I am the Deputy Director of the Bansidhar Institute and the founder of the Dominance Water Technologies Private Limited. I have been into the 23 years being as a professor at the Banasli Vidyapeeth. I have started my career from that place. I am the Chevening Fellow of the University of Oxford and CRISP Fellow also. And I have been into the like the visiting fellow and on the deputation at the Sevenci University, Turkey also. These all institutions has shaped my career, and I'm being the part of the DCM Shriram International Limited also. So I have been into the where I started my career. I established my lab in the molecular biology and the DNA fingerprinting, where I did a lot of work on the different molecular markers in the plant system, genetic diversity. And then I got the various good MHRD Center for Excellence. It is a MHRD grant, and where I established the Center for Excellence. There I initiated the water research in 2014, and with the help of the plant biotechnology, because I came from the plant background, so the development of the nanophytoremediation technology in the year like 2013-14, and the different technologies which relates to the different adsorbents, regeneration capacities, and how these toxicity levels of the nanomaterials affects on the plants. And luckily, all these 8 technologies are at the various levels of the commercialization. Today, I can say that where I have established my labs from the Banasli Vidyapeeth, where the spin-out company has also originated, that is the Dumlins Water Technologies Private Limited. And in the upcoming slide, I will show you that how my basic research has taken up to this level. Currently, I'm working on the interface of the biology, chemistry, and the physics also. And our labs are working on the 2D and 3D nanomaterials, soft materials, functional surfaces. Also, it's part of the life sciences and the plant biotechnology, but mainly into the chemistry emulgations. We are working on the device fabrications and all this, all these things can be converted into the valuable products and the technology that can be deployed by the industry or any startups also. So it is a story of various, like during my journey, I have mentored many, like 5,000 the female students and the 15 PhD students also. And I have also mentored various scientist positions who are working under me. So it is a story of the failure, frustration, triumph, and success. It is a nurturing, like the story of nurturing the various women scientists and the entrepreneurs, and the path is also going on. Still, it's not completed. And from this slide, I would like to take you the 20 years back when I started my journey from the Banasli Vidyapeeth, where I have seen that the surrounding area is like endemic to the fluoride problem. And although I came from a molecular biology background, but to solve this issue, and I have a very small dream that one day that whatever I am doing in the lab, I would like to see the impact in the real world. So I took this as a research question a way back, and I found that 200 million people globally and 66 million are suffered from fluorosis in India. And despite of this, so many of the like various options available at that time also, like the reverse osmosis, phytoremediation, ultrafiltration, ion exchange, and electrolysis. So what was the problem with the fluoride that could not solve at this level also, and it could not reach to the last mile? So there are various problems that I have discovered through my basic research, that most of the technologies are targeting about the bound fluoride, and they're targeting only when they're working with the standards, not working in the real environment, or when they are facing a lot of other combination permutations with the different ions in the environment. And this fluoride size is so small, ionic size, that it can pass through the RO membrane also. So I've worked on the basic sciences, and basic sciences with relates to the water also, with relates to the plant also. And I did the lot of work in the areas where that how can this fluoride affect the plants also, nanomaterials affect the plants, where it is accumulated. So I address every basic questions, that how to improve the heavy metal and the non-metal removal efficiency and speed up the process. Is the microbe, nanoparticles, or the metal affect the plant biomass growth or other parameters, and how to make this process sustainable? These was various simple scientific questions, and I did a lot of work in this phytoremediation aspects, and find out that by amalgamations of the 2-3 things like plant growth, rhizobacteria, hydrogels, quantum dots, and the nanomaterials. And one finding in my lab was very exciting. It is known as the copper carbon nanofibers. And these copper carbon nanofibers are giving a very good impact on the normal plants. So I took this idea and take up for the phytoremediation. And then I published in the Environmental Science Nano that this carbon nanofiber can act as a micronutrient carrier in plants way back. And then this like I have translated into the field, and I have showed this field tested technology, and the patent has been filed in the year 2019, and it has been validated with the help of the Krishi Vigyan Kendra also at the Banasli Vidyapeeth. So till now that I have published more than hundreds of the papers, 7 patents, 15 guided students, and H index is 39, and mostly the things like where the academia is working. But I thought that this is not the end of the journey, it is the start of the journey. And whatever we are doing, it should come to the last level. And it was the time when that our Indian ecosystem is also moving, and my dreams also shaping up. And there are two things where I have I want to sustain my talents of the raw talents, like the women scientists for the lab. And one day that one female scientist come in my lab and then say that, ma'am, I cannot go for the postdoc, please do something for it. And at this the same time, Indian ecosystem also going through the maturation and the direct grants are coming. So I thought that I should start something for the translation. And I got and to solve this issue for the fluoride and all these things, I started a very small idea, the startup company. So these ideas are coming through a very just normal, just when we are walking in the corridors, that one day that our research scholars came and discussing that how we can solve the leaching of the nanomaterials. And I given them idea that why we can't build a tea bags like pouches. And then we have just scientifically validated our idea. And this idea got wide media attention at that time without my any efforts. And this gives me motivation to do the translational research ahead. And I applied for the BIREC and luckily I got the various BIREC grants, and this idea also get various innovation awards as well as the fundings from the government of India. So the journey begins with a single step. And then I have established the Domini's Water Technologies Private Limited. And this is the team. One, Dr. Samina was my student and is the co-founder. And she has done 14 years of business development experience, and one of my co-founder is also Mr. Sujit, who was giving the 26 years of manufacturing and scale-up experiences. And the Domini's is working well with the come out with the many years of the research that the smart defluoridation nanorod and the one smart water purification unit for the community centers also. These are the technologies in which we have worked with the polymers, chemistry, as well as the catalysis. So these are the things that I have learned during my way, that whatever I have done in the plant biotechnology, but for the market, you have to develop a cost-effective and very compatible technology that can be scale up. So we have validated this product and the Rajasthan different, and we got an order from the 110 units from the government of University of Delhi also. And this is creating the impact, and we are into the phase of the scale up where I have joined hands with the DCM Shriram also. So this is the field deployment, जहाँ पे हम लोगों ने जो है फ्लोराइड की यूनिट्स को जो है फील्ड डिप्लॉय किया है, लार्जली वैलिडेट कर रहे हैं तेलंगाना के अंदर और राजस्थान के अंदर, ताकि जो है फाइनल स्केल अप के ऊपर जब जाता है तो ऑल द इंफॉर्मेशन वी कैन इंटीग्रेटेड इनटू द फाइनल यूनिट. एंड दिस यूनिट it can manage the fluoride, maintains the levels of the fluoride, as well as can remove the fluoride and not only fluoride but other heavy metals and microbes, and can give the purified water to the community levels. So when I was working at the Vanasli Vidyapeeth and was my different stages जो है मेरे सारे innovations की, then luckily these all are the TRL stages, and it took 15 to 20 years, and now it is reducing due to all AI and all these things. But the Shriram came at the right moment in my life, and I took this opportunity to create, to accept this opportunity to translate all my work to the field level as well as the for the industry deployment also. So this is the Bansidhar Institute, which is under the edges of the DCM Shriram International Limited. And this institute vision is to bridge the gap between the academia and the industry. And where I have also started my work to carry forward that one very good example for my basic research to the field deployment. And it is the lab scale to the pilot, commercial scale zinc recovery treatment of the wastewater technology. So all the industries are suffering from zinc sulfate, and 8 to 10 metric ton of sludge. जो वहाँ बनता है, it's a great challenge, and water recovery again to the manufacturing is also the biggest challenge. So to solve this issue at the lake of 3 lakhs liter water per day capacity, I propose the method which is the chemical, physical, and the biological. So my knowledge with the biological sciences, my knowledge with the chemistry, and then I learned how to integrate with the physical system. And then I propose a technology that can be translated, has been translated already, I can say, already you can say that I started my work with the 200 ml of water in the synthetic water. Then I started with 100 ml per day with the real water samples. It is a TRL 1 to 3 stage. And then it went to the 4 to 6 when I did all these things into the pilot of the batch mode as well as the continuous mode. And then now it is at the level of the 3 lakhs liter water per day capacity. Recently, the patents has been filed and it has reached to the TRL 0 to the TRL 9 stage. And recently it has been deployed at the industry, and the 3 lakhs liter water per day has been saved. So it can say that 10.8 crore liters per crores per year जो है volume है वो पैसे का बचता है, and it recovers 275 kg of the zinc sulfate per day, so around 2.97. So these kind of the things of the translation you can see easily. It's a story that where you have joined hands with the industry and you can show the clear-cut demonstration of the translation. And from the TRL to the TRL 9, I'm doing a lot of things with the industry partners, and you can say this is a DST water innovation project in which we have taken it for the TRL 4 to TRL 6. And this is my team members, and this is the pilot facility that we have established at the PDU Gandhinagar with our collaborators also. And this is very good project with the TDP that I am currently running. And it was my knowledge with the plant biotech, in which I have improved the shelf life of the different vegetables and the produces, and how it can help in the agriculture supply chains with the cutting edge cooling technologies. So recently this project has been funded by the Technology Development Board, and it is a proof of concept of the study that the CO2 can enhance the ripening, or can enhance the can reduce the ripening, we can say, and we can enhance the shelf life of the fruits. This is the containers in which the company DCM Shriram International Limited is working on. So we have integrated these two things. One is the CO2 technology with this container, and you can see that it's working so well, and it is now for the pilot demonstration, and like in the, I think like 5 to 6 months, it is completely commercialized the technology and can disrupt the market also. So this is the just we have faced the TDP midterm review meeting for that. I also created the, like how I just converted my lab to science and then to the society. I am involving in various awareness programs also. And through my own journey that I got various awards during this process. And recently I have been selected for the fellow of the Royal Chemical of Society. And during, I'm blessed that I got, but it's not my success. It's all my like teamwork that I would like to dedicate all these awards to all my people who has having faith in myself and work dedicatedly day and night for the very good cause. And these are the things that we have got awards for the our entrepreneurship also. And the story is still like we are doing it. So I can say like that it's not so easy to work and to identify your purpose of living. And on the day-to-day basis, I can say we have to face a lot of challenges in our day-to-day functioning. तो मेरा एक ही चीज है कि सारा कमाल दर्द का है, यूं ही कोई मिर्जा गालिब बना नहीं है. जब हमारा पाथ अलग है तो हमारा दर्द अलग होगा, और हमारा स्ट्रगल भी अलग होगा, हमारी कहानी भी अलग होगी. तो हैव अ फेथ इन अवर सेल्फ, फेथ इन द ऑलमाइटी कि द वन डे इट मेक ए सेंस कि जो आप कर रहे हैं वो एक दिन आपका जो है, यू कैन नॉट connect the dots backwards, but you cannot, but we can have a faith and we can see that it has a huge impact. तो इस वक्त जो है I have been fortunate enough, and I'm thankful to a lot of the people during my way, during my journey. And I would like to say that that to identify the purpose should be the purpose of living. And I'm very thankful to the Society of Plant Research that I'm truly honored. So I can say right now at my career at this stage that we need to bridge the gap between the academia, industry, startup, society, and government to make a real impact. So all our publications, all our patents should see the light of the day. Still, the millions of the people lack access to the safe drinking water, and industries are seeking the sustainable solutions, and our society also looking for the very good innovations. Now the time has come that we need to join our hands and we need to think out of the box and join the hands and bridge this gap. And I can say that my story is bridging the gap between the academia, industry, startup, society, and the government. And I'm living this on the day-to-day basis. And I hope that one day it makes sense. And this story still needs to go further, and miles to go is still ahead. So thank you so much, sir, for your kind opportunity to deliver this talk, and I'm looking for the long-term collaboration with the, like, how can I serve the Society of Plant Research, because my base is plant research, but now I'm doing the translation part, and I've integrated with all the life sciences, physical sciences, chemical sciences to make it a good purpose that I'm doing, that one day it makes a sense and it should serve the society in a large sense. Thank you so much.
Thank you so much, ma'am, for your inspiring and informative lecture. Now, moving towards the Best Doctoral Thesis Award lectures, I would like to invite Dr. Gaurav Jan from Amity Institute of Biotechnology, Amity University, Rajasthan, Jaipur, to kindly deliver his lecture. Dr. Gaurav Jan. Dr. Gaurav Jan.
Yes, ma'am, I'm here.
Okay, okay. You can start with your presentation, and kindly, it's a humble request that kindly stick to the time slot allotted, na? As we are already running out of time.
Okay, ma'am. I'm audible?
Yes, you are audible. You can increase your volume little bit, I think.
Now it's okay?
It's okay, it's okay.
My slides are visible?
Yes, your slides are visible.
Okay. Good evening, everyone. I am deeply honored and grateful to receive the Best Doctoral Research Award from the Society of Plant Research. I would like to express my heartily gratitude to Dr. Nagar sir, boss ma'am, and esteemed committee members of the society. I am Gaurav Jain, Assistant Professor, Amity Institute of Biotechnology, Amity University, Rajasthan. My doctoral thesis work is entitled on insect-inducible expression of pectin methylesterase from Vidania somnifera for broad-spectrum insect resistance. I will share some of the findings of my doctoral work, then I will share the achievements from the doctoral work. Next slide. I think slides are not moving.
Have you provided
Yes,
it in
yes.
the office?
Yes, sir, I have provided.
Okay, then it should be done from there.
Now it's moving.
It's visible. Yes, now it's moving. You can start with the presentation.
Okay. My work is basically on to protect the crop. Actually, national agriculture scenario, we have at least 50% of productivity we have in our hand, and the rest is lost at the pre-harvest and the post-harvest level. My work is to protect the crop at the pre-harvest level, and the main reasons for the pre-harvest level is the insects. There are various major agronomical insect pests are there, and different molecules are reported to protect the crop against a specific pest. But till now, there is not a single technology that can protect the broad spectrum of insects. We can say that none technology is available to protect both chewing as well as sap-sapping insects. So the idea is, do we have any natural molecules through which we can protect a large number of insects? And the answer is plant methanol, which is a natural molecules emitted by all the plants. It is produced by the demethylation of the cell wall by pectin methylesterase. So my work is to identify the most putative biotic stress inducible pectin methylesterase gene. And with the help of that gene, I'm going to overexpress it in the tobacco plant and to check whether the overexpression of methanol will help in protection of crop from the insect pests. So we have chosen Vidania somnifera as a source plant. Our lab report reported that in natural conditions, Vidania emits large number of methanol as compared to other family members. And from Vidania somnifera, we have identified 54 pectin methylesterase genes. And during the insect attack, we have checked the pectin methylesterase activity, which was increased up to 3 to 4 fold. And at the same time, we have also checked the methanol content and emission level. And it was shown that the methanol content and emission was also increased up to the four-fold high as compared to the normal plant. So next we have to choose which of the genes out of 54 is insect inducible. So we perform gene expression analysis and found WSPME 28 is the putative genes which is upregulate upon the biotic stress level. So this is the pictorial presentations upon the insect attack. The WSPME 28 gene was upregulated and it results in the overexpression of methanol from the plant. So we choose WSPME 28 as a source putative genes to express in the tobacco plants that we have cloned under the both constitutive as well as the inducible promoter system. And we have identified the stable transgenic lines through agrobacterium mediated genetic transformations. And we found in the transgenic plants, we have found 16 for up to 15-fold high methanol emissions in the control plant, in the transgenic plant as compared to the control plant. And the insect, then we have performed the insect bioassay against the both chewing as well as sap-sucking insects. In chewing insects, we have chosen Helicoverpa and in Spodoptera, and as a sap-sucking insects, we have chosen whitefly and aphid. And we have found around 85% of mortality as compared to the control plants. So till now, we have a gene WSPM28, which is upregulated during biotic stress. We have cloned it under the constitutive and inducible promoter system and transformed into the tobacco plants. And we have analyzed the transgenic lines showing up to 85% mortality against the insect pests with the 16-fold high methanol. The next question is, what is the fate of that methanol? Well, as methanol is a volatile organic compound, that volatile compounds impacts on the nearby control plant or not. So the next we have did a systemic analysis of systemic defense response over expressing PME lines on the neighboring non-transgenic plants. We have designed a chamber, a uniform airflow chamber, in which we capped a transgenic plant as a emitter plant and a control plant as a receiver plant. And a uniform airflow occurs from one end to the another. And we have collected plant samples at different time intervals level, like 2 hours and 24 hours. And at that time, we have performed transcriptomic analysis, and we found the phytohormone and signaling pathways were highly upregulated, like jasmonic acid, salicylic acid, calcium signaling pathway. So through these studies, we have concluded that WSPME 28 is the putative genes that we have overexpressed in the tobacco plant. And the transgenic plant shows up to 85% of mortality. And the next is the fate of methanol. Methanol also act as a signaling molecules in between the plants to plant communications. And the outcome of this work are several publications. Apart from publications, I have awarded Young Scientist Award, Best Oral Presentations, and Research Excellence Award from Kumaon University, Nainital. DST Avsar's Best Story Award. Apart from the publications and award, I have currently received ANRF Prime Minister Early Career Research Grant. This is the downstream work of my PhD. The project title is Deciphering Methanol-Mediated Interplant Communications through Integrated Raman Spectroscopy and Metabolic Approaches. And I want to acknowledge my supervisor, Dr. Praveen C. Verma, Senior Principal Scientist, CSR NBRI, where I have worked my entire doctoral work. My co-supervisor, Dr. Veena Pandey from Kumaon University, Nainital, UGC for providing financial assistance, JRF and SRF, and Professor Vinay Sharma, Director, Amity Institute of Biotechnology, Amity University, Rajasthan, for providing me an opportunity to apply in this award. Thank you.
Okay, thank you, Dr. Gaurav, for your presentation. And I would like to invite now Dr. Suresh Ramanan from ICR Institute, Jhansi Research Institute, to kindly deliver his lecture.
Is my screen visible, ma'am?
Yes, sir.
Okay.
Your screen is visible. Yes, sir.
Thank you. Respected dignitaries, seniors, and colleagues. Thank you for this opportunity. I would like special thanks to the founders and the president, madam, for giving me this opportunity and recognizing my work. My work titled as Ecological Prospecting of Genus Melia in India. So the research question I was trying to address through my research work was, Melia is one particular genus which has been a challenge and a myth for the taxonomists for quite a long time. But as an application-oriented scientist, when we look Melia, it has a close association with the neem, Azaractica. So these two genus has been very much productive in terms of among the Meliaceae family. But when we take this Melia alone, there is a recent hype on one particular species called Melia dubia, commonly known as Malabar neem, for its multi-purpose utility, because it can be utilized as paper and pulp, it can be used for plywood industry, it can be used for small timber industries. So such a versatile species depending on the age of rotation. So it offers us a broad market, and it's an indigenous species. So that sparked the entire forestry, agroforestry and plantation sector to go around the species. But when we look into the background of this genus, there are three species: Melia dubia, Melia composta, Melia asatica in India. But all these three species are considered to be synonymous or non-synonymous. Only recently there was some taxonomical work as well as genetical work to differentiate Melia dubia as well as Melia asatica. But there is a third species called Melia composita. So there is some sort of confusion among this particular genus. So what we did is we wanted to understand the entire facet of this genus using empirical, morphometrical as well as genetical characterization resource techniques. So we addressed two important questions. One is whether all these three species are same or different, and given the hype and promotion of particular Melia, what it is suitable location where it can be cultivated or what it will be its future. So to realize this things, we had three major objectives in our mind. First is to understand the global research focus on genus Melia. So we used bibliometric approach as well as science mapping approach. Second, we have used characterization technique for morphological as well as ecological characters of all the three Melia species. As you can see the photographs, different Melias in different parts of the country. The third is forecasting a potential area for cultivation, which is done by niche modeling. So the methodology which I adopted, overall, I tried to use an open source data sets or open source softwares so that it is replicable. First is bibliometrics. So we have used bibliometric R packages for the morphological traits. The traits are listed as there, and it is a simple work. But when we come for niche modeling, we try to go for the conventional Maxent software using Java-based interface. For niche overlapping studies, we tried Wallace software based on R platform. So first results will be understanding the global perspective of genus Melia across the world. So when we see four predominant species comes into picture, one is Melia asatica, Melia dubia, Melia vulcansi, Melia composita. Among this, Melia asatica is a species spread across all countries. I hope you can see the names of the country here. But whereas in the case of Melia dubia and Melia composita, only India has the major contributor of researchers in terms of literature. So that clearly indicates that these two species are confined to India when compared to Melia asatica. But the specific case of Melia composita is only India and Saudi Arabia as the two countries where research papers are originating from these two countries, indicating that Melia composita is highly specified to Indian context. So further, we collected samples, fruits and seeds, and did a morphometric analysis, and we did germination studies to understand how the species behaves after it grows up. The fruit length and width was a deciphering characters to differentiate the species. Melia dubia was size bigger compared to Melia composita and then Melia asatica. When we did a niche study to decipher the species with 33 samples for morphometric location, we had more than 280 field location of different age group plantations of Melia. And based on that, when we decipher 19 bioclimatic parameters, we could see Melia dubia is a little bit an sensitive species. It cannot tolerate cold. The minimum temperature is 20 to 25. Whereas Melia asatica and composita is a species which can thrive in coldest region, and also it can withstand much drought condition. This was the current niche condition of the species. When we did a niche overlap, we can see despite the morphological dissimilarities between Melia composita and Melia asatica, the niche overlap was there, indicating that Melia composita and Melia asatica are more or less having a same distribution in the northern part of the country, and particularly some parts of northeastern India. So from a climatological perspective, in future climate, how this species will behave. And we see Melia dubia, which is having the current height. We can say that this particular species will have a decline in area over a period of time owing to its vulnerability in terms of climatic parameters. Whereas Melia azadirach and composita can withstand the vagaries of weathers, and the predictions across different SSPs and different models, both the models indicates that the area or a highly suitable area is keep on increasing. To summarize it out, what we can or what my study has presented here is there was a taxonomical uncertainty with regard to species, and now Melia composita being highly confined to India alone, this as a species has to be, or whether it's a taxon level work has to be furthermore scrutinized, because India is the only country having research on that particular species. The second part is all the three species has distinct characterization in terms of morphometrics. So when we say Melia composita is under question, morphometrics indicates that there is a variation. So in the future works, we can understand how it has evolved, how it can, genetical variation is there, genomic level studies or phylogenetic studies can carry out in future. In terms of ecological niche modeling, that is to applied orientation, where we can grow these species, we can see Melia dubia will decline. But that species has been proven to utilizable in a short rotation of 4 to 5 for paper and pulp, 7 to 8 for plywood industries, and more than 15 years for small timber. So that is going to be get affected by our climatic conditions or climatic future scenarios. So these other two species, especially composita and azadirach, if we can do an interhybridization or an interspecies work, some sort of things can happen out so that we can tap it out on a fast-growing indigenous species rather than relying on exotics like eucalyptus and casuarina. So with that, my study is complete, and we could do some good publications. 4 research papers have come out, 2 conferences and 1 book chapters, and 1 more paper is under process. So that is the work which I was able to complete during my PhD. And I would like to thank my organization, my guides, as well as Mizoram University's people who are supportive enough to understand my work. Being a pan-India study, there were a lot of people who helped in my field surveys and other things. I would like to recognize all their contributions here. Thank you, one and all.
Okay, so thank you for your presentation. And now I would like to invite Dr. Lokesh Thakur from CSIR IHBT, Palampur, Himachal Pradesh, to kindly deliver his lecture. Dr. Lokesh Thakur.
Okay, thank you, ma'am. Am I audible?
Yes, you are audible. You can start with your presentation.
Okay, wait a second, ma'am.
Okay.
Ma'am, is my slide visible?
Yes, your slides are visible.
Okay. So good afternoon, everyone. I am Dr. Lokesh Thakur, currently working as Senior Project Associate at CSIR IHBT Palampur. First of all, I would like to sincerely thank the Society for Plant Research for giving me this opportunity and for selecting my thesis for Best Doctoral Thesis Award. It is truly an honor for me. So in next few minutes, I will be presenting my PhD work with you, which was entitled In Vitro Selection of Ginger Tetraploids for Tolerance Against Rhizome Rot. So coming to the background of my research, ginger is a monocot diploid crop having chromosome number 22. Its underground part, which is called rhizome, is used for consumption in various industries and is also used as a seed for next season propagation. Due to its vegetatively propagation mean only. So there is a major constraint of rhizome rot disease, which is caused by the combined effect of Fusarium and Pythium species, which leads up to 70 to 80% yield loss. So there is a great need for its genetic improvement. But due to some limitations like low genetic base and vegetative mode of propagation, the conventional breeding approaches are not that much effective. So we have to go through the unconventional approaches like mutation breeding, chromosomal doubling, or genome editing, etc. So in our study, we used the combination of chromosomal doubling and in vitro selection techniques for development of improved lines and then evaluated them at morphological, physiological, biochemical, and molecular level. This is the basic hypothetical model of our research, where we first developed the tetraploid shoots using colchicine from the diploid shoots of ginger and simultaneously prepared culture filtrate of fusarium and pythium and subjected the tetraploid shoots for in vitro selection on MS medium. Then the tolerant shoots were hardened and evaluated at multiple levels. So there were three major objectives of my thesis, where I had to select the tetraploid shoots against culture filtrates of these fungi and then evaluate them at in vivo conditions, and finally their biochemical and molecular characterization. So before beginning the in vitro selection trial, we wanted to confirm that the tetraploid shoot that we have generated are actually in their original tetraploid state, as many studies have reported that the artificially developed tetraploid shoot sometimes gets reverted back to its original state, which is diploid in this case. But we got positive results with flow cytometry and chromosomal counting data. Simultaneously, we prepared the culture filtrate from the pure cultures of fusarium and pythium and tested it for the toxicity in vitro by inoculating the ginger shoots in the culture filtrate. Simultaneously, the in vivo pathogenicity test was also carried out, where the mass culture of fusarium, pythium, and their combination was spread around the hardened ginger plants. And after a few days, the symptoms were clearly visible, which were further validated by microscopic analysis, and the presence of spores confirmed the pathogenicity of these fungi. So for in vitro selection treatment, various concentration of fusarium and pythium culture filtrates were mixed in the MS medium. For fusarium culture filtrate were at 20% concentration and pythium at 11%, and their combination at 15% and 7.5% pythium culture filtrate showed 3.3% of plant survival after third selection cycle at 20% FCF, and 1.67% shoots survived at 11% of PCF concentration. These shoots were further multiplied and used for further evaluation. So after the selection, the three different types of selected shoots along with diploid control and tetraploid controls were hardened till their maturity and used for various evaluation, morphological, physiological, biochemical, and molecular evaluation. There were some reports which said that in sometimes the in vitro selected shoot does not show equal amount of tolerance in the in vivo conditions. So we wanted to confirm it. So we used fusarium and pythium crude culture for this purpose. In case of diploid plant, the symptoms were visible after few days, but in case of tetraploid control plant, only the leaf margins got yellow. And in case of selected tetraploid plants, there were very little to no symptoms at all. Therefore, confirming that the tolerance has indeed been translated to in vivo conditions as well. So then we went for the morphophysiological and biochemical evaluation. This is where in the figure at top left corner, we can see this is the ginger plants, which is control and the selected lines after harvesting. We got the difference in the leaf size as artificially induced polyploidy enlarged this cell size. So we can see here that the tetraploid control and selected tetraploid leaves were bigger as compared to the diploid leaf. Similar was the case with the rhizome. The rhizome size was also smaller in case of diploid as compared to tetraploid and selected plants. And in stomatal level, we also, due to smaller size, there were more stomata and in the microscopic field view. And in tetraploid and selected tetraploid, there were small, lesser stomata, but bigger in size. So the physiological parameters which were carried out by infrared gas analyzer also showed the physiological activity was higher in case of selected tetraploid lines as compared to diploid and tetraploid control lines. The 6-gingerol content, which is 6-gingerol, which is a phenolic compound that is often associated with the antifungal activity, level was also higher in case of selected tetraploids as compared to diploid and tetraploid control plants. Other than this, the antioxidant enzyme activities like catalase, superoxide dismutase, and peroxidase was also tested, which were also elevated in case of the selected tetraploid as compared to control plants. And the MDA level, which are the indicators of lipid peroxidation, were found to be higher in case of control plants, as the selected tetraploid plants were better in handling the antioxidant scavenging, ROS scavenging. Then we did the molecular evaluation of selected plants with the various types of marker like Scott marker, genomic SSR, EST SSR, and their combined SSR markers. In each dendrogram, we found that the control diploid plants were separated into a separate cluster, indicating that there might be some genetic changes in the tetraploid and selected tetraploid plant, which are making them different from the diploid plants. The population structure analysis also revealed that there are a total of 5 population combination in the selected tetraploid lines. We got some interesting results with the GIN1 and GIN9 markers when genomic DNA was amplified. We got some unique bands in certain plants, and we eluted them out and sent them for sequencing. Upon sequencing, we got to know that these are related to the disease-resistant protein-like genes, therefore confirming the tolerance at molecular level as well. After that, we wanted to check the expression analysis of various genes using QRT-PCR, like resistance gene, terpenoid biosynthetic pathway, antioxidant enzyme, and gingerol biosynthetic pathway genes. These are the relative expression as compared to a diploid control plant, and in almost all the genes were upregulated as compared to diploid control plants. This heat map is also created using the same data, and here we can see the selected tetraploid lines are showing upregulation of most of the genes. These are the research outcomes. We were successfully able to generate stable tetraploid ginger lines with enhanced tolerance to fusarium, pythium, and their combined culture filtrates. This is the subsequent field trial that we conducted last year, and this year also we have sown the seeds in the field, and these are the 2 publications that came out of my PhD work. And I also got Best Poster Presentation Award at 47th Symposium of Plant Tissue Culture Association India held this year at NIPGR in New Delhi. And finally, I would like to acknowledge Department of Biotechnology, Dr. Yashwant Singh Parmar, University of Horticulture and Forestry, Nauni, Solan, Himachal Pradesh, and CSIR IHBT Palampur. And special thanks to my advisor, Dr. Manisha Thakur, for always helping me. And my lab members, Dr. Keshav, Dr. Deepika, Dr. Pooja, Dr. Vivek, Dr. Madhur, Amanpreet, Akanksha, Riddhi, and Anshika. And finally, once again, I would like to say sincerely thanks Society for Plant Research for providing me this wonderful opportunity. And thank you for listening so patiently. Thank you all.
Thank you, Dr. Lokesh, for a wonderful presentation.
Thank you, sir.
And now I would invite Dr. Shreya. Yes. Dr. Jyoti, are you here?
Sir, actually, my mic was mute. I was unable to unmute it. That's why. Yes, sir.
Okay.
Okay.
Please.
Yes, sir. Okay, Dr. Shrija. Dr. Shrija from Central University of Haryana.
Am I audible, ma'am?
Yes, you are audible. You are audible. You can start with the presentation.
Ma'am, can you see my slides, ma'am?
Yes, your slides are visible. You can start.
Okay. Good evening, all. I'm Dr. Srija, currently working as a National Postdoctoral Fellow from at Department of Biotechnology, Central University of Haryana. I have done my bachelor's and master's in botany from Kerala University. And I have done my PhD in biotechnology from Punjab University under the guidance of Dr. Humaira Sana and Dr. T.R. Sharma. So my PhD thesis was understanding evolution of tonoplast intrinsic protein tree and its role in plant seed development. So basically, tonoplast intrinsic protein is an integral membrane protein belonging to aquaporin gene family. It can selectively transport water and other small solutes. Tip structure resembles an hourglass model formed of 6 transmembrane alpha helix interlinked with the 5 loops. Importantly, the solute specificity of tips are determined by 2 constraints. One is the LPA motif present at loop B and loop C. Another one is the aromatic arginine selectivity filter consists of 4 amino acids. So previous studies showed that it can transport water, urea, ammonia, glycerol, and hydrogen peroxide. However, what TIP3 transport was unknown. Also, the evolution and role of TIP was also unexplored area. So I developed these objectives to address the knowledge gap regarding TIP evolution and functional role. My first objective was to identify and understand the evolution of tonoplast intrinsic protein homologs in different species. Here I have done genome-wide analysis over 100 different plant species, followed by comprehensive phylogenetic and evolutionary analysis. My second objective was to study the tissue-specific expression of tonoplast intrinsic protein subfamily. Here I conducted RNA-seq-based expression analysis across multiple plant species. Additionally, RT-qPCR analysis of TIP3 was performed across different tissues, seed developmental stages, and germination stages. And my final objective was to study the role of tonoplast intrinsic protein 3 in seed development and germination. Here I cloned and characterized soybean TIP3 gene in yeast expression system and identify its solute specificity and also done tobacco infiltration for subcellular localization of TIP3 gene. Further, to understand the functional role, I have knocked down the TIP3 gene in arabidopsis via CRISPR-Cas-based genome editing. For this, first of all, I have developed the RNA, then I developed the construct, and then I transformed this construct into the arabidopsis plant through agrobacterium-mediated transformation via floral dip method. So the evolutionary analysis, revealed that TIP family originated in bryophytes and include a distinct subgroup known as TIP6. Interestingly, TIP6 exhibit greater similarity with the hip subgroup of aquaporin, suggesting that it may have evolved from hip-like ancestral proteins. Later, this TIP6 during the course of evolution expand and diverge into 5 groups. Notably, the TIP3 evolved in seed-bearing spermatophyta with a strong seed-specific expression. For instance, the expression analysis showed that TIP3 expressed in seed. Also, TIP3 showed a gradual increase in expression during seed developmental stages, and also it shows higher expression at the onset of germination, suggesting its role in seed development and germination. We also done a molecular dynamic simulation and understand the pore structure of TIP3 protein. Further, in yeast assay, we clone and characterize soybean TIP3 into the yeast system. So here we can see, that the GM TIP3 showed repressed growth compared to the empty vector in media containing hydrogen peroxide and boric acid, suggesting that TIP3 can transport these solutes. Further, we also used tobacco infiltration method to identify the subcellular localization of TIP3 and find that, as the name suggests, it's found in the tonoplast. Also, it is found in the vacuolar membrane and endoplasmic reticulum. Further, through CRISPR-based genome editing, showed that knockdown of Arabidopsis TIP3 resulted in altered pod color, reduced pod size, and changing branching pattern. These findings suggest that Arabidopsis TIP3 play an important role in plant seed development there. So to conclude, tips evolved in seed-bearing plants with a strong seed-specific expression. It can transport solutes such as hydrogen peroxide and boric acid. Mutation in TIP3 cause altered pod size, its color, conforming its role in seed development there. So therefore, TIP3 serves as a promising target to improve seed vigor and crop productivity. So I have a total of 27 publications with an H index of 13, and these are my top 5 publications come out of my research thesis work. And I have also filed a patent on transformation protocol of mustard. And some of my major achievements include National Postdoctoral Fellowship by ANRF and Best Oral Presentation Award and Best Poster Presentation Award for my thesis work. And I have also got JRF and GATE. And this is my research group, and I would like to thank SPR for giving me this prestigious award and also giving me the opportunity to share my thesis work with you all. Thank you.
Okay, thank you, Dr. Shrija, for your excellent presentation and sharing your research contributions. Now I take the privilege to invite Dr. Rahul Kumar Goswami from Magdeburg, Germany, to kindly deliver his lecture.
Hello, ma'am, I am audible?
Yes, you are audible. You can start with your presentation.
Hello.
Yes, you are audible.
Hello.
Dr. Rahul? Hello, Dr. Rahul?
Hello.
Hello?
Hello.
Hello? Hello?
Dr. Rahul, you are audible.
Yeah, thank you.
Yes.
My slide is visible, sir?
Not yet. Yeah.
Now it's visible.
Okay. Okay. Good evening, everyone, and thank you for giving me opportunity to present my work and awarding me for the best doctoral thesis from this Society of Plant Research. I, Rahul Kumar Goswami, currently doing postdoc research at Helmholtz Center for Environmental Research, UFZ Magdeburg, Germany. I completed my PhD from Department of Microbiology, Central University of Rajasthan, India, under the guidance of Professor Pradeep Verma. Today, I am going to present and highlight my key finding of my PhD research, that is development of low-cost cultivation system for enhancement of microalgae biomass production. Sir, my slide is moving.
I am unable to see your slide. Now your slides are moving. You are at introduction slide.
Yep, yeah.
Okay.
So let me introduce about the microalgae, that are the diverse group of aquatic photosynthetic microorganism present in different kind of aquatic environment. It's classified as the eukaryotic cells and the prokaryotic cyanobacteria. The benefit of the microalgae is can fix the CO2 from atmosphere and remediate the wastewater from environment and maintain the environmental sustainability. Their biomass contain different kinds of biomolecule which have great application in the different industrial sector like biofuels, functional food, agriculture, pharmaceutical, and cosmetic. And this way, it's maintaining the principle of circular bioeconomy. So, and the problem and rationality of my study, as we know that The energy shortage, burning of fossil fuel, shortage of functional food, agriculture issue, and the water shortage are the major global challenges facing today. So microalgae, one of the organism that have the capability to resolve this kind of issue. So despite their wide application, their commercialization is still limited due to the undeveloped biorefinery, high cost of cultivation, and the high cost of harvesting. So we can optimize this growth and condition by optimizing the light day-night cycle and temperature and designing the suitable cultivation system. But their large-scale study are very limited due to the less adaptability of algal strain in a changing environment. So this is my summary of my PhD research work. Ma'am, it is visible now? Hello?
It's visible. It's visible.
Yeah. So uh, based on the literature, I selected two microalgal species. One is a Picochlorum and another is Tetraselmis indica. These two algal species are fast growing, able to tolerate different environment and the conditions. And I work in Rajasthan where the temperature and salinity are very high. So that's why I selected these two potent microalgal strain. And then I find their cultivation mode, and then I design low cost cultivation media using the raw municipal wastewater and the secondary treated municipal wastewater with the combination of conventional ASN3 media. Then I optimize their growth condition, and after getting the result, I use the media for further experimentation and then develop a co-cultivation of Picochlorum and Tetraselmis in two-stage sequential cultivation system. In this two-stage cultivation system, first stage we use raw municipal wastewater 75% plus 25% of uh, conventional media and cultivate the 2 ratio of tetraselmis and 1 ratio of picochlorum. Then after the first stage of cultivation, the treated water and the mixing with the 50% of conventional media, and then we cultivate 2 picochlorum ratio and 1 ratio of tetraselmis. After getting the result, we scale up to the sub-pilot scale and study the HRT. Then we also study the impact of seasonal variation on their growth in outside the environment. Then after getting the successful result, we scale up this process in the 1,000-liter open pond system. In each set of experiment, we monitor the growth, nutrient removal performance, biomass, and biomolecule productivity. So these are the major outcomes of my PhD study. As you can see there in the first graph, this is laboratory scale the data. Here the co-cultivation ratio shows the much higher biomass in first and second stage of cultivation compared to their monoculture. And the similar trend was observed in a sub-pilot 10-liter cultivation system. Then we only scale up the co-cultivation ratio in a 1,000-liter system, and we found that it maintained the biomass productivity as similar to the laboratory condition and matches the result 85% of the outcome from laboratory conditions. Then we also determine the lipid content in the lab. It's more than 45%, and which is also higher in the co-cultivation compared to their respective monoculture, and similar trend was observed in a sub-pilot scale. In the large-scale cultivation system, a slight reduction of lipid was observed, but also it is a comparative good compared to the other conditions. So we also did the FAME analysis of lipid, and the co-cultivation 2TS1PC contained the highest FAME content, and which was higher to their respective monoculture. We also study the nutrient removal efficiency of microalgae and compare with the monoculture and co-cultivation. In lab scale condition, it removes the 85 to 95% of nutrient load from wastewater, and in the first stage, and it's ranging between 70 to 82 in the 1,000-liter system. While in second stage, the co-cultivation shows 85 to 100% of nutrient removal approximately, And this in 1,000 liter system, it goes 80 to 92%. As here you can clearly see, this is a raw wastewater, and this is the first stage treated wastewater. And after cultivation, this is the result of co-cultivation, and this is the result of monoculture. As you see, the co-cultivation shows the better clearance of the wastewater. These are some photobioreactor which I designed for microalgal cultivation in my PhD journey. These are my research outcome related to my PhD work. I also filed a patent which was published, mass scale sequential microalgal co-cultivation system for enhancement of biomass production and biomolecule production. So it published in December 2024. These are my research profile. Till date, I published 30 articles, 11 book chapter, and 1 patent. And most of my publication are in Q1, Q2, and Q3 journals. I also got the more than 205,000 citation with H index of 22 and I-10 index of 32. I rank among the highly ranked scholar prior to 5 years as the top 5. I'm also the associate editor of Journal of Applied Biochemistry and Biotechnology, Springer. I also got the 2 times for travel grant award to present my research at international stage. And I also got the best poster presentation award in 2023 at 64th annual international conference organized by AMI, Society of India. So I would like to thank Professor S.K. Bhatnagar, Professor Vandana Bose for awarding me the best doctoral thesis, and the all other member of Society of Plant Research. And I also like to thank my PhD supervisor for nominating me for this award and motivating and inspiring me to do good research throughout my PhD journey. Thank you.
Okay, thank you, Dr. Rahul, for your valuable presentation and your research, for sharing your research perspective. May I now invite Dr. Sama Subramanieshwari from National Institute for Research on Commercial Agriculture to kindly deliver her lecture. Yes, you are audible. You can increase your volume up, like it's little bit low.
Okay. Ma'am, my screen is visible. Hello. Hello.
Yes, it is visible. But yeah.
A very good evening to everyone. Myself, Sama Subramanieshwari, Dr. Sama Subramanieshwari, pursued PhD in Department of Genetics and Plant Breeding under the guidance of Dr. Saikat Gandhait. I'm here to present my PhD research work on the topic in vitro direct regeneration and induced mutagenesis for enhanced production of major strival glycosides. Imagine a natural sweetener that is 300 times sweeter than sugar, helps diabetic patients, and has anti-cancer potential. Yet its commercial cultivation is still limited because of poor propagation and low metabolite production. My research aimed to solve both issues simultaneously. This is outline of my presentation, which includes why I have chosen this particular crop stevia, key significance, methodology that I have executed, result, and key achievements from my research work. Coming to stevia, it is a perennial herbaceous medicinal plant, belongs to family Asteraceae with chromosome number 22. It is known as sweet leaf or candy leaf because of the presence of diterpene strival glycosides, majorly riboside A and stevioside. These are believed to be 350 and 250 times sweeter than sucrose. Due to sugar-like and various therapeutic properties, it grabs immense commercial attention. However, conventionally propagated plants produce inadequate strival glycosides. There is a possibility of enhancing strival glycosides via in vitro mutation. Researchers are finding to develop elite clones enriched with higher strival glycosides. As I mentioned earlier, the demand for this natural sweetness among Indian farmers and pharma companies is increasing day by day rapidly. but the food and drug industries are not able to attain such targets due to poor seed germination and inadequate clonal propagation. Keeping this in view, there is an alternative, that is in vitro mass multiplication and value addition via in vitro mutagenesis. These two options can act as an alternative to meet the requirements of growers and pharma industries. On this backdrop, this work eventually aimed at to optimize neoteric PGR formulation mediated enhanced direct regeneration system, acclimatization, fidelity assessment of stevia, and the second objective is to develop new variants of stevia with enhanced stevial glycosides using gamma rays mediated induced mutagenesis. In case of objective 1, we have surface disinfected shoot tips of BSR 2021 and inoculated in 5 different cytokinin in three different concentrations to assess their impact on shoot multiplication. Later, the proliferated shoots were inoculated in rooting medium for another 4 weeks to develop complete plant life. This is biphasic protocol, whereas shooting is followed by rooting. In case of monophasic protocol, we have used liquid medium, polyamines, and growth inducers to reduce the span. In this way, we have developed a complete plantlet. Finally, the in vitro regenerated plantlets were transferred for primary acclimatization, followed by secondary acclimatization. The acclimatized plantlet was assessed with the mother plant at various levels at morphology, cytology, flow cytometry, molecular markers, and HPLC analysis. Here are some very interesting results that I have obtained in my research. Here we can see the maximum shoot multiplication was noticed in 1.5 metatoplin. Maximum root formation was noticed in IBA supplemented media. Even in the control, we have found roots because it might be due to endogenous hormone levels. The acclimatized plantlets showed 95% survival rate. Via seam-in cross sections of acclimatized plantlet with the mother plant, it was proved that growth and development was normal. When it comes to molecular marker analysis, we have obtained 100% monomorphism with stable ploidy level via flow cytometry analysis. Here we can see that the peaks in the flow cytometry, the peaks in the flow cytometry retain, proving that there is no genetic variation from the acclimatized plantlet to the mother plant. As I mentioned earlier, to further enhance, we have used liquid medium and different polyamines and growth inducers. Among different polyamines, we have found that 200 micromolar putrescine produced maximum shoots with maximum elongation and leaf count within 4-week period. The work of metatoplin induced and polyamine mediated were published in 2 different journals in South African Journal of Botany in 2 different papers. After establishing a reliable regeneration protocol, we moved for metabolic enhancement. 2-week old microplantlets were exposed to different gamma irradiation doses to optimize LD50. To the optimized dose, 500 cultures were exposed and advanced to M1V1 generation. The M1V1 progeny is recovered from the mutagenic treatment were advanced to M1V2, as it is established fact that M1V1 generation is not stable. Hence, we have moved to M1V3 as well. In the M1V3, we have selected 5 stable variants, and these are moved to M1V6. Among them, 5 variants, they were moved to M1V6 generations. Variants were analyzed at morphological level, histological level. They were estimated at flow cytometry, HPLC analysis was carried out, and they were acclimatized. Here we can see the impact of different gamma ray doses on in vitro shoots. Here we can see the gradual and morphological decline of trait-specific differential decline of in vitro growth rates with increased gamma ray dose. Via probit analysis, it was found that 18 gray was optimized dose in case of stevia. Here we can see that comparative growth performance of variants with the control. In case of morphological differences, BSR V1 exhibited superior growth performance, followed by BSR V2, having narrow needle-shaped leaves. Here we can see that comparative difference of the variants to the control at M1V2 generation as well as M1V6 generation. In case of BSR V1, the internodal elongation is less, still it produces maximum lifts. But in case of BSR V5, the internodal elongation is more, but it produces less number of leaf count. When it comes to semithin cross sections, it was found that the variance exhibited significant variation in comparison to the control. Via three different molecular marker assessment, it was found that the variance exhibited 4.6 polymorphism comparison to the mother plant. From the flow cytometry analysis, it was revealed that the in vitro regenerated variants showed pattern different from the control, but with identical ploidy status. There is no change in the ploidy level. When it comes to main key finding, the HPLC chromatograms, here one can see the HPLC chromatogram of ribadicide is standard in comparison to the control BSR V1 and V5. Below, you can see the HPLC chromatograms of Stevaside, control BSR V1 and V5. It was found that it was recorded that there is 1.6 and 1.7 folds higher ribadicide A and Stevaside in BSR V1 variant. Here we can see that the mutants were successfully acclimatized under ex vitro conditions. Here we can also observe that there is a significant variation in BSR V1 in comparison to the V2 and the control. HPLC analysis revealed that the BSR V1 and V2 produced 1.5 and 1.8 folds higher ribadicide A in comparison to the control. The identified mutants was approved for all India trials in 5 AICRP centers by ICA DMAPR at 22nd Annual General Meeting. The work of mutagenesis paper was published in 2 different articles. One is Sugar Tech with NAS rating 7.9 and another one is Plant Cell Tissue and Organ Culture with NAS rating 9.0. To conclude that, from the present work, we have established an efficient and reliable regeneration protocol. Gamma ray mutagenesis successfully enhanced the stevial glycoside without altering any ploidy level. The developed variants has strong potential for agriculture and industrial application. Finally, I would like to acknowledge Society of Plant Research for giving me this opportunity, and Bidhan Chandra Krishi Viswavidyalaya and DAE-BRNS for the funding and support. I would like to thank my PhD supervisor, Dr. Saikat Gantayat, and Dr. Co-supervisor, Dr. Jayati Mazumdar, Dr. Somnath Bhattacharya, Dr. Sudhir, and Dr. Suchita for their support and guidance throughout my research and my collaborations. Thank you.
Okay, thank you. Thank you so much for your presentation, Dr. Sam. So with this presentation, we have covered all the award presentations. And may I now take the esteemed privilege of inviting Professor Ramaswamy Nanna sir, President South Zone, to kindly deliver the concluding remark and bless the SPR fellow awardees with his inspiring words. Sir, we are deeply honored by your gracious presence, and I humbly request you to address the gathering and share your valuable blessings with the awardees. Sir, over to you.
Sir, unmute कर
Sir,
लीजिए mic.
kindly unmute yourself, sir.
So, sir, kindly unmute your mic. There should be a symbol of mic. You click on it.
Yeah.
Oh, fine.
Now it is okay.
Yes, sir. Yes, sir.
So, good evening, everyone. My dear young and dynamic scientists from different institutes and universities. At the outset, I would like to congratulate all the SPR Fellow Awardees 2026, and especially I would like to thank the founder chairman of SPR, Professor Bhatnagarji, for this lecture series program and also assigning me to say concluding remarks. All the fellows presented their research nicely, which are inspiring and interesting on various topics. All are having a brilliant caliber. Really, I appreciate on behalf of the SPR and on my own behalf for your excellent research carried out. But my suggestion and advice on this occasion is to support and encouragement for further growth and development of our SPR by newly awarded fellows. Once again, I congratulate the newly awarded fellows. Finally, I thank the organizers, especially Professor Bhatnagarji and Vandana Bose, for giving me this opportunity. Thank you, one and all.
Thank you very much, sir, for your inspiring blessings and encouraging words for the awardee. Your visionary thoughts and valuable guidance, along with your motivational remarks, have truly inspired the young. Have been inspired the young researchers, scholars, and participants present here today. So now, with the permission of chair, sir, should we conclude today's session?
Yeah, a few words I would like to speak before we conclude.
Yes, sir. Yes, sir. Sure.
It was a wonderful session, and I'm very much thankful to our national president, Professor Vandana Bose, and our South Zone president, Professor Ramaswamy Nana, for gracing this occasion and sparing a lot of their valuable time despite their so many assignments. Making presentation by the fellow awardees and the best doctoral thesis awardees was mandatory, and they were supposed to present, but the presence of senior executive body members is an add-on privilege for the society. They were present in order to bless the fellows and the best doctoral thesis awardees with the vision that these young awardees, they will be instrumental in making good research contributions to the journal Vegetos, which should be of very good quality, of global standard, so that their journal and their society should be established globally. Though it is already global, but your very active input and contribution in the form of research papers and by making very quality review of the papers if they are assigned to you, because you are a part of this family now. You are the new entrants to SPR family and to the Vegetos family. So being the new entrant, it is a big responsibility on the shoulders of all our young fellows to make potential contributions for the global growth of the journal. As you already know, it has already completed almost 38 years of its existence since 1988. And we are planning to celebrate 40 years of its existence next year.
Oh, okay.
In Delhi, most likely. And then your potential and your contribution, your support will make it a memorable event. And I'm sure that more responsibility lies on the shoulders of our young fellows, because we have served for 40 years since its insertion in 1988. When I established the society, long back, almost 38 years back, it was a vision that it should be a global society, a global institution, and that dream has now come true. It is being published, the journal is being published by Springer Nature, as you know. It is a Scopus indexed already, and we are moving ahead to be included into Web of Science very shortly. because we have cleared almost all major steps for its impact factor next year. So with these words, I thank our National President, Professor Bandna Bose, our Chief Patron, Professor R.R. Hanchinal, our South Zone President, Professor Ramaswamy Nana, and all our executive body members and editorial board members, previous awardees, past awardees, who joined this very important webinar today and yesterday for making it a big success. Last but not least, I would thank Dr. Jyoti Singh, who had been my student, and I naturally depend on her for many activities of the society. So I thank Dr. Jyoti Singh for being with us throughout yesterday and today to moderate this very important webinar. Thank you all very much and wish you a very good luck. We shall be in the process of preparing citations, and the process of conferring these awards will be in process right from today onward. Thank you all very much once again. Thank you.
Thank you so much, sir, for your kind words and blessings. Now, on the behalf of Society for Plant Research and the organizing committee of SPR Fellow and Best Doctoral Thesis Award Lecture Series, I extend my heartfelt gratitude to all the distinguished dignitaries, eminent scientists, academicians, researchers, scholars, and participants who joined us today and yesterday from the different parts of India and abroad. We sincerely thank all the awardees and the speakers for their highly informative and enriching lectures delivered during today's and yesterday's sessions. Their scientific contributions and experiences have truly added a great value to this prestigious lecture series. And I would also like to express my special thanks to Professor Dr. S.K. Bhatnagar, sir, Professor Bandna Bose, ma'am, the organizing committee, screening committee, technical support team for their dedicated efforts and constant guidance in making this webinar successful. With this, we conclude Day 2 SPR Fellow and Best Doctoral Thesis Award Lecture Series. Thank you once again for your gracious presence and participation. Wishing you good health, success, and a pleasant evening ahead. Thank you. Thank you, everybody.
Thank you. Thank you.
Thank you. Thank you very much.